this one? Or?
Yeah, this one.
This is fine.
We'll directly take audio.
Good morning all. So I welcome you all for the 7th day GIAN workshop. Hope you all enjoy. So today, as part of the lecture, mine will be talking more about the chloroplast transformation. So it's an application based topic. So have you heard about this topic or have you somewhere read about this topic earlier? So this is more about, my lecture will be more of interaction. So I expect you to answer and we go ahead like that. So have you anywhere read about this chloroplast transformation or do you have any idea about this chloroplast transformation? Okay, no problem. Then it's more easy for me. I will be, so you can just follow me whatever I'm going to say. If you have any questions, you can stop me and you can ask me. Fine. So I'll be asking a few questions in the introduction slide. So do answer that. So these many days, I'm sure that there are many resource persons who are talking about the chloroplast genome. But any plant or algal as a whole system, if you take, can you say how many genome does it has? Generally. Chloroplast? Oh, I thought you will miss the mitochondria, but you are clever enough there. Yeah. We always forget about the mitochondrial genome, but even mitochondria has a genome. So the nuclear genome everyone might be knowing about. This slide I made it based on the chlamydomonas, but still the nuclear genome is about very long. It's about it has a 17 linear chromosomes and it has a genome of 17,000 genes it's code for. And in terms of the chloroplast genome, it's circular. As yesterday also we seen about it, and it is about 200 kilobase pairs, and it codes for roughly around 100 to 110 genes. And mitochondria is very smallest one among the three, and one important feature, interesting feature about the mitochondria in terms of chlamydomonas, it is linear, whereas in other higher plants, in other organisms, they are circular. So that point has to be noted down in terms of chlamydomonas. And as it is a smaller one, most of the genes are transferred to the nuclear genome, and it codes only for roughly around 8 to 10 genes. That's it. So I'm sure that the structure you might be continuously seeing in all these 7 days of lecture series. No? Even in your dream, I think you might be getting this structure. But in this structure, something is missing. This is a very template structure of the chloroplast, but something is missing in this structure. Can you say what it is?
Where the genome is located.
Can you say what?
Maybe the location of genome.
Yeah, genome is not there. So most of the general template where we got it from the chloroplast, no, we forget to identify there is a genome. So I'll be showing you in the next slide. In the upcoming slide, it has a multiple nucleon genome. So this is a plant cell. Generally, a plant cell will look like that. And where is our chloroplast in terms of size? They stand in the cellular level. We can see it's about 1 to 2 micron range, in between 2 micrometer range. And an average plant cell will stand from 10 to 100 micrometer. And we can see the atoms are 0.1 nanometer. So in this scale, we can clearly see it's very small, right? And inside, so our talk today is going to be very in-depth. And all these thylakoids, everything is a very ultra microstructures we have been studying these many days. And yesterday you guys did the thylakoid isolation also. So, and this also you might be knowing, the product of the chloroplast, whatever we breathe, whatever we eat, both comes from the chloroplast, right? O2 and sucrose, as well as energy for the plant itself, ATP and NADPH, comes from the chloroplast byproducts. So now we'll come to our topic, we'll stick to the topic, what is transformation? Now I need some answer. So let's try to simplify that term. Chloroplast, I think everyone knows by now. So transformation, what might be the synonym for transformation?
Taking off foreign genetic material.
You're directly into that, okay. Okay, I just need the simple synonym for transformation. What do you generally mean transformation?
Change.
Change something. So the terms literally means you're changing the chloroplast, right? So is it something like this, you're going to change the chloroplast physically? Like a superhero or something, or something like a, are you going to change the structure of the chloroplast? No, right? So we are going to change, then what is actually this transformation? So we are, as, brother, what's your name?
That's okay.
Yeah, now you can explain that. What is chloroplast transformation? Not from there, whatever you know. You can,
Okay, that means
you can use the microphone. You just press.
like example of any foreign DNA or foreign gene into chloroplastic DNA.
Yeah.
We can say that's the example of chloroplastic.
Yeah, exactly. We are just trying to incorporate our gene of interest into the, particularly the term has been highlighted, into the genomic of the chloroplast. In other term, plastome, we can say. And stably expressing those gene of interest. For what purpose we will be generally doing this? Why do you want to integrate the foreign gene into a, particularly a chloroplast? It's like for studying the gene function, or for engineering to produce more product or more production of the metabolism, whatever you are expected, the gene of interest. That's what, right? So here we can clearly see the three-dimensional structure. We can clearly see the nucleus, the chloroplast nucleus, and it will be having multiple copies. It's not like one or two. It will be having, depends upon the organism to organism, the number of nucleoids will change. So that point has to be considered. The chloroplast nucleus is multiple in number. Okay, now I'll be again coming back the difference between three genomes I have mentioned, nuclear, chloroplast, mitochondria. What are the, first let's see the nuclear and chloroplast. What are the difference I have mentioned clearly. The first, in terms of integration, the mechanism by which the foreign DNA is integrated is different. So I hope you will be knowing about the recombination happens, the two things. One is non-homology based and another one is homology based. So what happens in non-homology based? Any idea? In non-homology based, it's a very rapid process. It doesn't require any homology sequence. Homology means a complementary sequence. So it doesn't require. For example, when this is generally a repair mechanism. Whenever a DNA in the host get damaged, the host system try to repair that, right? So in order to repair, they follow two mechanisms. One is homology based, another one is non-homology based. In non-homology based, it doesn't require, it doesn't see what is around. It directly joins without finding any complementary sequence or not. It's a very fast, but it's also error prone. So in that process, few bases will be deleted, as well as few bases can be inserted also. So this nuclear is a random process. Nuclear insertion will be a random process. So once you create any double-stranded break using CRISPR, any method, the sealing will be very fast. So if you give any foreign material without any complementary sequence or homology arms, still they can able to incorporate them. Whereas in terms of the chloroplast transformation, they follow homology based. So here, whenever there is a breakage, it doesn't seals up directly. It looks for homology arms, like a sister chromatid. It looks whether there is any sister chromatids or complementary sequence available nearby. So that so that I can complement it without any error-free process. There will not be any deletion or any addition. So they are more precise in terms of repair mechanism. So that's why chloroplast transformation is more targeted, they used to say always. And in terms of expression, again they are variable. In terms of chloroplast, they are highly stable. What might be the reason, do you think? Variable in nuclear. First of all, I think Tripathi sir, yesterday he was clearly explaining about the plastic genome versus nuclear genome. So plastic genome are mostly prokaryotic based origin. So it doesn't have any introns. And introns are not there, you can avoid the euchromatin, heterochromatin regions will be avoided here. So do you hear about this term position effect variegation? Position means the expression of the gene depends upon where it is inserted. Means if, for example, if your gene of interest, you are inserting in a highly active region of the genome, that is called euchromatin. obviously the transcription will be very active. Whereas in case if they are incorporated in the heterochromatic region, where it is transcriptionally less active, then generally the expression is going to be down-regulated. So why this problem comes in the nuclear transformation? As I mentioned already, it follows non-homology based. It's a random process. So we cannot assure it will be exactly inserted in your expected region, or sometimes there may chance to incorporate it in the heterochromatic region also. And the possibility of introns also there in the nuclear. Whereas in the chloroplast, those things were not there. That's why they are high and stable. Whereas in terms of the nuclear, they seems to be variable. And silencing mechanism common in nuclear transformation. Silencing, have you all know the silencing? Means you would have, yeah, you can tell.
Means you can't express for some reason. We can say that
Yeah. We have physically inserted the gene of interest, but they cannot able to do the function. Means either they are transcriptionally silenced, translationally silenced, depending upon which kind of mechanism they have incorporated. Why the silencing happens? Any idea?
like one method is RNA interference.
Yeah, perfect. One of the method is RNA, but why it happens, RNA interference?
Means if the homologous sequence of a specific RNA can produce protein. If there is a homologous sequence of that RNA is present inside the cell, then that from the whole.
No, you're telling about the methods, but I'm telling why it happens. My point.
It's a defensive mechanism.
Okay, why? Defensive against?
It's a foreign, they will detect it.
Yeah, exactly. Whenever you insert a foreign genetic material, the cells also we have immunity, right? The cells also feels that okay, a viral load or something came in, so I should protect myself. So they activate. These are one of the mechanism in order to prevent them from the host any damage from the viral side or any other pathogen. This they will consider the gene of interest you are giving. Sometimes the host may consider them as a transgene or a viral pathogen, genome of origin. So they try to silence by activating RNA interference. And another possibility, as I mentioned, euchromatin, heterochromatin. And another thing is methylation. Methylation. Sometimes they try to stop the transcription by methylating the promoter region of the gene. So all this silencing is possible in the nuclear, but doesn't in the chloroplast. Why? Again the same prokaryotic origin, whereas they are eukaryotic. So we can see the advancement, how the nature has evolved from prokaryote to eukaryote. What are the defense mechanism it has incorporated in the nuclear genome. So we can also compare these two things by looking at the graph. And inheritance, already you might be knowing, it follows the Mendelian, whereas here it follows the maternal inheritance. So what is the advantage of maternal inheritance? It will be straight. It will be generation after generation. It will not be spread through pollen. The GMO, that problem will be overcome in terms of the chloroplast transformation. Whereas in nuclear transformation, that laws are very strict for them. So people consider, okay, this is more safe, but still they have some limitation, which I will be talking in the last slide. And methods I will be talking. The majorly used for nuclear transformation is electroporation, glass beads. And here the ballistic particle current bombardment methods are very more prominently used. So now the third transformation. While preparing this slide only, I had the thought, why mitochondrial genome has not been considered? They should also be, till now, because I was not much familiar with mitochondrial transformation. Are you anyone familiar? I heard about nuclear transformation, chloroplast, but mitochondria somehow they are avoiding. Why? Then I just went through, check what is the reason. The first thing they mentioned is the barrier. Even chloroplast is too much difficult to penetrate for the foreign DNA, but mitochondrial is very tough. They're telling it's more rigid, impermeable for accepting any foreign DNA, and the accessibility for the mitochondrial genome is further difficult than chloroplast. In terms of chloroplast, they are telling they are suspended in the matrix, whereas here they are telling they are embedded inside the matrix. They are telling. So it's the accessibility for the nucleoid is very difficult. That's they are telling as a one point. And another point they are telling the recombination is very poor. in terms of the mitochondrial genome. Whereas there they use homology, right? Here the recombination efficiency is lower, so the success rate is further down. And yeah, they are telling by itself the genetic are not ready to take in more foreign genes. They are like resistance to take foreign genes when they try different things. And finally, there is no selection markers. This selection, what is the selection markers? I will be talking in the upcoming slides. They don't have any proper selection markers here for the mitochondrial specific nucleoid. And already I have mentioned one of the point to study why we are doing the transformation is to study the function of the gene in the nuclear. Chloroplast we already studied by creating many insertion. Whereas in mitochondria, as I said, only 10 genes are there. So those function has already been studied. So people are thinking why to struggle more when there is no requirements are there. So that's why mitochondrial transformation is less popular. But still there are reports. Few has been done in some carrots. I have found some reports, but they are not big journals. Seems like very small works. I'm not sure the percentage of success. If it is a very successful, then by now it should have been a hot topic. So I found these are the reasons for them not to be more popular than the chloroplast and nuclear transformation. So let's see how they have evolved. Like any techniques have been started at one point, and slowly gradually they increase and adapt different features, and they'll just evolve and they'll come back. No? So let's see. This chloroplast transformation has been introduced in the year 1988 by Boyan et al. So he did the chloroplast transformation in Chlamydomonas reinhardtii, and he used the marker that is pectinomycin, which is specific to the chloroplast genome. So there 1988 they started, and from then they moved to the higher plants. In 1990 reports are there for chloroplast transformation in higher plants, particularly in tobacco. And they also proved that the chloroplast transformation inherit through maternal. So that important point has to be mentioned. In 1990 they have identified this maternal inheritance. And then they identified promoters. As I mentioned, till this the expression is there. There is a transient expression. But once they identified a specific promoter for the chloroplast, they can able to upscale the expression level. So when they use PSBA and Rubisco large subunit promoters, the expression increased to a large fold. So that is very important, no, for chloroplast transformation, because we want to upscale whatever product or whatever gene of interest we are going to clone. We expect them to express to a maximum scale. So this is also one of the important tasks. 1994 they have did it, and in 2000 they moved to non-model plants like potato, carrot, lettuce. They did this transformation, chloroplast transformation, in the non-model plant system also. And then they moved, in 2010, they started using this method for producing vaccines, other byproducts, like a chloroplast bioreactors. They have started using the chloroplast as a bioreactors. And till now, there are many multiple techniques evolving. One of the techniques is CRISPR. And CRISPR is generally localized for the nucleus. Now they are slowly adopting or trying to adopt to the chloroplast transformation also. But the success rate is still not that efficient. And because CRISPR mostly for nucleus, even I have did the CRISPR transformation for nucleus. But chloroplast specific Cas9 need to be designed, and that is still challenging, and the work is still going on. Hope in the future there will become. So this is the overview of the chloroplast transformation process. So generally it has a vector, and in this vector we need to design your gene of interest, and I will be telling you about what are all the parts a vector should have in the upcoming slide. Once you need to design the vector, and there should be a transformation method to transfer this vector into the organism of interest, and once they are transferred, integration will happen inside, and then you will go for the selection in the antibiotic, whatever the antibiotic genes you have selected, and finally you will be confirming using screening using PCR, anything. So this is the workflow for the entire chloroplast transformation. Any doubts till now?
Which vector is used?
There is no any specific vector. I will be telling about the vector. No. You need to construct one particular vector. Any vector, it's not like specific like PUC19, PCA, there is nothing like that. You can use any one, but it should have the following component. Components are very important. I will be telling, and have you all aware of the textbook definition of the vector, right? What is the textbook definition for vectors? Mike. I can't hear, or audience cannot hear. Molecular vehicle. She's telling molecular vehicle. Definition should be elaborative. Yeah, it's partially correct, but you can elaborate a little. It's a extra chromosomal circular DNA. That is one point. It's extra. It's not the part of the genome. It should be extra chromosomal circular DNA capable of self-replication. They should also able to replicate by itself, not dependent on the nuclear genome. So that itself defines what are all the components it requires, right? So it requires promoter. Basically, it requires promoter, multiple cloning site, origin of replication, selection markers. So if you want to know what are all the individual components, you can ask these questions. Like first, what is selection marker is used for means you can ask the question, how can I differentiate transformed versus non-transformed cells? So answer is, if you have a selection marker, you can able to select, right? So that's the use of the selection marker. And for the origin of replication, you should ask the question, how to multiply the plasmids. Okay, I have a plasmids, I made the construct, everything. But you have a single copy, it's not going to help, right? You need to bulk it up. So how you are going to do? So for that, you need a origin of replication. And for promoter, okay, everything is done. If you transform, there should be somewhere to start and stop, right? Promoter and terminator. So that's the promoter, you need to keep the promoter. And multiple cloning site, obviously where you are going to insert a gene of interest. That should have restriction sites. And this promoter, as I already shared you, they have discovered chloroplast specific. So promoter varies to where you are going to express. It's like spatial dependent. You cannot use one common promoter. And depends upon what is your purpose of expression, you need to change your promoter. Fine? Shall we move? Okay, this is an example of a chloroplast vector itself. I have taken it from a paper. And you can clearly see, as I mentioned, this is the origin of replication is here. And ampicillin resistance for in the E. coli in our selection marker for a prokaryote. It's not a selection marker for the chloroplast. Selection marker for the chloroplast is present here. This is our construct. This is a vector we can generally buy from any company available. But this part you need to be careful what you are going to do. So this part you can see this is a promoter for the selection marker. AADA is nothing but spectinomycin resistant gene. So 5' UTR, 3' UTR, followed by the gene of interest. For that also you should be having a promoter, 5' UTR, gene of interest, followed by the 3' UTR. So this entire construct is from your part. You need to design. You need to design specific to your interest. You want to keep this gene or any other gene, depends upon your organism which you are going to study, and what gene you are going to insert, which place they are going to express. All these matters. So once you design the construct, you need to develop the vector, either by incorporation using cloning, or you can recombination, many recombination method available, or you can synthetically buy the vector by saying the requirements. I need these following components. The company can make and give for you. So it's up to you, however you can make. Once this vector is ready, then you need to go for next step is transformation. Before getting the transformation, how this transformation method has been developed. So for that we need to go back about the natural genetic engineering. So I think everyone would have know about the agrobacterium tumefaciens, right? So it's a soil bacteria or what it is? It's a soil bacteria, right? So whenever a plant, it causes what disease? Crown gall disease. So it's like basically it's a tumor. It induces tumor in the crown region, and as a result they can go and inhibit in the region, and they will take the carbon source from the plant. So this is a good example for how naturally an organism can transform their gene of interest and make use of the host translation mechanism and utilize the food resources whatever the host has for itself and survive. So you can clearly see this is the agrobacterium and this is a plant cell. So whenever there is in terms of root cells any damage, as you know the acetoserogenic molecule, there is a phenol, will be released, and this will be sensed by the virulent gene. By the way, I forgot, this also has a plasmid. You might be knowing the TI plasmid. TI stands for? Tumor-inducing plasmid. So like we constructed plasmid, they do have components like origin of replication they have, virulent gene they have, tDNA. tDNA is like the construct we made, right? Similarly, we can consider tDNA as our construct for time being, I can say. So this tDNA is the one which is going to be integrated. And virulent gene is the one which sends the, okay, there is an opening, we can get inside. There is a signal, I got the acetylcysteine. So virulence A and G will sense the acetylcysteine, and then it will activate the many virulent genes you might be knowing. So virulent T1 and D2 will cut the tDNA from the plasmid, and once they are released from the tDNA, with the help of virulence B, vir B gene, they will form the secretory type pyridine. So as a result, they can pass through from the agrobacterium and they enter the host plant. So how efficiently they form the passage and they enter. Once they are able to enter, again the virulence E will come to protect them from the nucleolytic attack. So they encode a gene which knows how to protect from the host organism. So virulence E will protect them, and virulence F will help them to integrate into the host organism. So transformation is done. So this is what we are also expecting now. We also expect our plasmid should enter but transform into the chloroplast, not in the nucleus. That's the difference. And once they are entered, they make use of the host translational mechanism, and what they will make 2-3 products, right? Cytokinin, auxin, that creates the tumors, right? And opine is used as a carbon source, and they live. So this is this is how it happens, and how scientists make use of this method. So they just, someone asked, stop.
It's about plant cell, right? What about a plant?
Yeah. I'm just starting and giving an overview. It's not like this method may not be applicable for everything. But even reports are there in Chlamydomonas, but not in many plants. They are very, the limitation of the agrobacterium mediated transformation, they are not universal. They have very narrow host range. Even today I just read the paper, they are dicots. Yeah, that's what. There is a report in Chlamydomonas when they incubate this agrobacterium and they added acetosyronjoline, they are still able to infect. So that seems to be interesting. But the percentage of success was still under debate. It's not complete. So what the scientists decided, okay, everything will keep the same, virulent gene, origin of replication. Only they are going to change the tDNA, because that is the part which is going to be transferred inside the DNA, genomic genome, right? So here, this is the actual one present in the DNA, see tDNA, the auxin, cytokinin, this is open metabolism genes. So here, the right border, left border is nothing but the homology based sequence. So everything is same, only the in between, whatever the genes which is code for tumor as well as for the carbon source, they replaced with whatever your gene of interest. And they make the same construct, and now they make them to infect. So they found that the infection was successful and they could be able to transcribe and translate their gene of interest very successfully. So this is the basis for whatever the modern transformation we are doing. This is the basis for whatever transformation we are doing. That's what I want to say from this slides. Any doubt? Yeah, now we will go to the actual methods of chloroplast transformation. So, biolistic or particle gun bombardment. So, have you heard about this ballistic missiles? Ballistic missiles in war zones, they used to
split into
No. Ballistic missiles are nothing but they throw the missiles in a very high speed. In any war zones, they used to say they used to say ballistic missiles. So like a high projectile is something but it goes and drops like this, right? So they project the missiles in a very high speed. That is called as ballistic missiles. So this biolistic also comes from the similar term. We are just projecting our gene of interest in a particular microcarrier that can be either tungsten or gold. We just code our plasmid or DNA of interest, and we'll allow them to project in a high speed to hit our cells, algal cells or plant cells, so that they can able to penetrate the cell wall and they can able to deliver the DNA to the chloroplast in your organ. So that's how the name has been originated, and I have already mentioned the principle. The DNA will be coated with the microprojectile and accelerated at a high speed. It will be done in a specific instrument, gene gun. They call as gene gun. So the principles and the procedures are same. We need to coat the micro projectile. Micro projectile will be commercially available. You can buy. Initially they use a plastic micro projectile when they started this experiment. Then they developed slowly and they found that gold and tungsten seems to be very ideal. Right now they are using gold and tungsten. And they fire these particles in the helium pressure. They mentioned 400 to 1000 psi. So that high pressure they are pressurizing to fall so that they can able to penetrate. And finally, and any process, once the selection will be on the antibiotic, whatever the selection markers you give will be used for the selection pressure. They used to grow them for 1 or 2 generation, and from that they will be conforming finally using the PCR. So this is the process of the whatever I have said. They have flow chart we have mentioned. For example, let's take the plant, and they will excise the tissue. They will excise a single small tissue. They will cut the leaf into small pieces. And now my doubt is whether they, for this biologistic method, will they use cells or protoplasm? I haven't said, but you can predict. Or you would have read all these things before. Understood my question, right? Whether for this biologistic method, will they use a protoplast or the cell, active cell with the cell wall or without cell wall? Protoplast. How many of you say protoplast? Can raise your hands? Okay. Majority. How many says then who are silent? Can I consider you as a cell? Sure? Okay. It's more ideal to take the cells. Why? Because when we are going to use the high pressure, the mechanical damage will be very high. And when we are going to use the protoplast, again the recovery will be more serious damage to the protoplast, and recovery will not be that good. So when you use the cell, the recovery is quite possible. So this is the only method. The second method I will be talking, and that will be protoplast you should use. Whereas for this, you can use the cell, intact cell, that will give more better results. So we will be taking the cell, and this is the particle gun bombardment setup. I will be showing in the next slide also. So from the top, with high pressure, we will be settling the micro projectiles. Like a missiles only, I can say. It will be very high pressure. They will be going and hitting the cells kept at the bottom. So let's take 100 cells are there. Out of 100 cells, we cannot expect all the 100 will be a transformer. There may be a 50 cells will have a transformer, and out of the 50, hardly 5 to 6 will regenerate. Regeneration also very important, no? If they cannot able to regenerate, no use. So hardly 5 to 6 cells will regenerate in the selection media. We will immediately transform them to a selection media, and we can found hardly 1 or 2 or 5 shoots will come out. And from this we will be transferring to the root induction media. So and then we will going for the PCR confirmation to see whether at a specific location they have been inserted or not. So that's the importance of the PCR. It should not be randomly inserted, right? Sometimes chances are there. You should be very specific. Okay, this location they have been inserted. So for that only we will do PCR to confirm. Okay, this is the actual image of the gene gun. You can clearly see. This is the pressure knob. From the top, you will be throwing the micro projectiles, and you will be keeping your samples here in this location. So they will be with the high pressure. They will be penetrating. This I have explained. So advantage. This is like universal, quite universal compared to other methods like agrobacterium. I said it's like narrow host, and the next method I will be talking, that is a peg-based method. That is also very narrow range of host only you can be used. But whereas in this case, bialistic, it can go for any host, like any plants. It doesn't have any restrictions because it's just a physical. We are just introducing them. The regeneration comes in the next step. So this is most successful method in terms of the chloroplast transformation, and it enables a direct delivery into the chloroplast. It's not like the next method will be PEG, where you will be introducing the DNA in the cytoplasm. From cytoplasm they need to enter into the chloroplast. That is more difficult. Whereas here you are directly penetrating with the high pressure, no? So it can directly penetrate the chloroplast membrane. So the advantage of reaching the gene to the nucleus is more faster. And finally, the transformation efficiency is higher in the particle gun bombardment method. There are obviously limitations should be there. So requires the equipment, the cost, commercial point of view, we need to buy this instrument, and physical damage, as I have mentioned you earlier. it does physical damage. So the recombination and regeneration efficiency, it varies to species. But ultimately, if you see compared to other method, biologistic method is the ideal one till now, till date we can say. And again, the random penetration depth, it's again a random process, right? It's like we are just penetrating. Some may go for the nucleus, some will go for the chloroplast. This is common for any transformation method. So you cannot see this as a major negative for this method. But still, I'm just pointing out there is a chance it may also enter the nucleus also. So finally, there have been efforts have been made to directly deliver the proteins, RNAs, RNPs. What is RNP? Yeah. What is actually RNP? Yeah. The term itself mentioned it has a RNA and protein. So it's nothing but CRISPR-Cas9 has been evolved in a way. Initially, how they designed a vector which has a Cas9 gene, coding gene, and your guide RNA you will be coding into it, and you will be transforming. And inside the host, they will make the guide RNA and the Cas9 protein. They will form the RNP. So whenever the guide RNA combines with the Cas9 protein, this RNA and protein is called as RNP. So you know about the CRISPR working methodology, no? Not sure? Okay. So CRISPR works basically, the guide RNA is the one takes the Cas9. Cas9 is an endonuclease that can able to chop off the nucleus, but it cannot randomly do. It requires some guide sequence. So that works has been done by the guide RNA. So once this RNP is made, RNP I already said you combination of the guide RNA and the Cas9 protein. So this RNP complex will be taken to the nucleus depending upon the Cas9 signal. Every Cas9 has a localization signal. So for now, most of the Cas9 are localized to the nucleus. So they will be taken to the nucleus, and based on the guide RNA sequence, they will make a NIC. So similarly, what scientists are trying to do, chloroplast specific RNPs they are trying to develop. Means externally we can make a guide RNA which has a complementary with a chloroplast, a unique sequence which should not be present in the nucleus. And Cas9 you can anyway make it from, we can synthesize externally, and externally we can send them. As a result, they can directly enter. I can say the advantage of this method is duration is reduced. The transformation is much faster in this method. But still, it's all a question because already for getting a DNA itself a tough job. How they are going to send the RNP is again it's a very unstable molecule. So this questions has to be answered in future. These are all the how scientists works progress in the direction I just want to show here. So you can note down that particle gun bombardment is one of the universal method for chloroplast transformation. Next method I have been telling you is the PEG mediated transformation. PEG stands for polyethylene glycol. So it's a polymer. I personally used this molecule for some other my experiments, but it can be used for the chloroplast transformation also. So the principle behind it is when you incubate your gene of interest, the plasmid, along with the PEG molecule and your host stage, let's take algal or plant cell, but in this case you need to remove your cell wall. Cell wall barrier will not allow them to enter. Okay, so you need to use either cell wall deficient strains in terms of algal, or in terms of if you are going for a higher system, you need to use the protoplast only. And when you subject these three things, that is the DNA, PEG, along with your host, the cells, that is a protoplast, they will make the plasma membrane more permeable. In the process of dehydration, they will remove the water. In this matter, the permeability, I think while removing the water content or something, the permeability increased. Maybe because of the osmolarity change or something, they are mentioning the permeability of the protoplast or the plasma membrane increased. As a result, this particle can be able to penetrate them. And again, once they are entered, it's a random process. We don't know how many the DNA is going to enter the chloroplast, we are not sure. How many they are going to enter the nucleus, we are not sure. But more success rate in the algal system than the plant system. Why? This method is more successful in algal than in the plant system. Why? Yeah, because single cell and in terms of, let's take in Chlamydomonas, 70% is occupied by the chloroplast. So it's very close to the membrane also. So the chance of hitting the chloroplast is more in terms of the algal rather than in the plant cell, which has many obstacles because it is very small component in plant cell. But in algae, it is the major. So obviously the chance of entering the chloroplast is more compared to the higher systems. So I think that's what I mentioned. They require to use a cell wall deficient either by creating autolysin treatment, enzyme treatment, or you can use a mutant which doesn't have a cell wall. And these things I have already mentioned. Mix the plasmid with the PEG, incubate briefly, and finally the selection process will be same for everything. Any method, the selection process will be same. Let's go to the advantage. Method is very simple, no? You don't need any instruments. Very inexpensive. Just you need a PEG, remaining things your DNA, that's it. Very inexpensive. And the physical damage is not there here. We are not giving any physical damage. So that seems to be the advantage. But again, the limitation is cell wall. You need to generate a cell wall strength. It's not like you can use the entire cell. It's very much important. Cell wall strength you need to use. And the transformation efficiency also, here it is lesser. Because it's a random process, again I said there you are incorporating physically. But here we are not sure how many minutes, how many particles is going to get inside. These are all question marks. But still, as I mentioned, in algae this works. And finally, they are algal specific, not friendly to the higher plant system. So now we'll move into the selection markers. So selection markers are mostly antibiotics, antibiotic resistance gene they use. So the first one I mentioned already, they have identified or developed this method using spectinomycin resistance gene, that is nothing but AADA gene, which confers resistance to two antibiotics, spectinomycin and streptomycin. They used in Chlamydomonas and tobacco, these two. And next is APH, that gene they use, that is conferring resistance to the neomycin, and it's used in tobacco. And third one is APHA6, that confers resistance to kanamycin, amikamycin, they use in Chlamydomonas. And next is BADH, that gives resistance to betaine aldehyde, used in tobacco. And final one is SHBNE, zeoxin, used in Chlamydomonas and cyanobacteria. So these are all important antibiotics markers they have used. Still, it's expanding and it has to improvise as the day goes on. Because, for example, if you are using one gene, it's fine. When you are using multiple genes, if you want to transform multiple constructs, then the requirement for using more selection markers also increases, right? So selection markers keep on increasing, but this again gives a limitation in the GMO. What might be the limitation for using selection markers? Antibiotic resistance, you know that, right? So antibiotic resistance, for example, now all these are fine. In case if they are using this chloroplast transformation to develop any food crops, let's take in future, and they are going to have this selection markers which is antibiotic resistance. So obviously there is a chance that the antibiotic resistance seems to transform. So again the GMO constraint comes into the place. I will be talking this in the limitation part, just I want to give a glimpse about it. So this is the overall view about the entire chloroplast transformation, how it works. And as I mentioned, the construct you need to develop, that's a very important point, which has a gene of interest along with the selection markers. And you can use, once it is done, you can use any selection methods, transformation methods. Either you can use the bialistic method or the PEG mediated method. And this is how the recombination happens. The homology sequence I mentioned, no? It should be placed in between the right flanking region and left flanking region. And let's take, this is the plastic genome. And they found the complementary and they flip and they insert the gene of interest in the place that the homology region is there. And it becomes exactly in this region, the gene of interest and selection markers will be incorporated. Now, and then we will go for the selection markers. And now the problem is, as I already mentioned, in terms of chloroplast, you have only one chloroplast. In terms of Chlamydomonas, only one chloroplast. But in higher plants, as already the previous lectures you might be came to know that there are many chloroplasts. So there the problem between heteroplasmic and homoplasmic will come. But in my case, I use Chlamydomonas, I don't get this problem. But when you are going for the higher plants, definitely out of 100, again chance probability of 5 to 6 might be transformed, remaining all will not be transformed, right? So again the problem has to be overcome by growing them in selection media. Like this is again time consuming process. We don't know which stage exactly or it will turn to be homoplasmic. At least 6 to 7 generation, you need to maintain them in the selection markers to confirm that everything, every entire chloroplast, plastome genome has been transformed. So finally, once you are successful, the chloroplast transformation has been achieved, then you can go for confirmation with the PCR and you can release them. So now let's see the future, how this chloroplast transformation or researchers, how they work in this direction. For now, I have read the paper, there is a direction, future direction. They work on editing tools, how to improvise the editing, plastome editing tool, which are specific. As I mentioned already, once I was mentioning, the CRISPR-Cas9. Most of the CRISPR-Cas9 available are localized to the nucleus. So now they are trying to develop the Cas9, which can localize to the chloroplast as well. So that's the main thing. And DNA-free editing using RNP, that also I mentioned, or directly synthesizing the guide RNA and protein, and is it possible to send from externally, so that to reduce the time point in the transformation. That way they are thinking. And synthetic recombinases. Recombinase is the one which plays an important role in the recombination. So they can able to create a synthetic recombinase that can improve the efficiency of the recombinases. In that direction also they are thinking, particularly to create any point mutation. Is it possible to create point mutation in the plastic DNA? They just want to study that. And this is very interesting for me. They are trying to generate the entire plastic genome. Is it possible to design a plastic genome externally and send it to it? The idea seems to be very interesting, no? You can generate an entire plastome with all the genes of your interest, not for the plant, for you. Like, for example, in this case, they are telling carbon fixation, improving the carbon fixation ability, like improved, better efficient Rubisco. Calvin, what is the limitation of Rubisco? Any idea? Rubisco has one limitation, major limitation. Okay, another one major. They have affinity for CO2 or O2? O2. Okay, they have more affinity to O2, right? Compared to CO2. So in these kind of natural problems can be overcome. They are thinking in that perspective. Okay, how can I structurally design the proteins or the entire nucleum of our interest rather than only inserting a small part? Why don't we generate the entire nucleum of our interest? That's the entire plan. So new metabolic pathway, biofuel, lipids, whatever, fatty acids. It's like a programming. What I want, you give. Like that. Biosynthetic pathway for microbes, everything. And this is obviously expanding beyond the model species. Right now all the experiments they're not went to the cash crops, food crops. Their aim is to transfer this to the food crops. Is it possible? And chloroplast as a biofactories. This is what they want to do. So whatever they want to produce, is it possible to produce in the chloroplast? The advantages due to why they want to do chloroplast as biofactories, as they have mentioned, high copy. Instead of one genome making whatever product you want, 100 genome makes at a time. The value or the number will be more, right? So that's what they want to make the plastome. And gene silencing is not there, so constitutively you can express. Whereas in the nuclear genome, that hurdles will be there. And finally, containment, maternal inheritance. So whatever the genes they have designed will be constrained in the organism only, it doesn't spread to neighboring or any other food crops. So this is the future application. In short, the major groups they want to work in agriculture, in terms they want to create a better crop, in terms of climate resilient, better nutritional, anything they want to invest in the agriculture. And they were pharmaceutical, as I already mentioned, they want to utilize them in the biofactories, whatever the vaccines, everything they want to produce in them. Even in the space biology, they are trying to implement in terms of oxygen evolution, whether they can use this organism for generating the oxygen in the space, for at least for the astronaut usage, not for the whole, for the astronaut usage, whether possible. And climate mitigation effect also, like they can generate organism which can take more CO2 to reduce the environmental CO2 level. Finally, anywhere I need to conclude with the limitation. No? So I brought the limitation of this chloroplast transformation. So any method, it does has limitations. So we need to understand them and we need to use them in a better way to overcome this limitation. That's the note of bringing here. So as I mentioned, the limitation is species range. It's still under the level of the model plants. It doesn't move to the commercial crops. So once they move to commercial crops, there will be more drastic changes in the society it will be created. But for now, these limitations are at present day limitations. It may not be the future. Future will change for sure. They may use the limitation and they come up with a better idea. So for now, the species range are very narrow. So that has to be converted, or they have to improvise they should increase the species, the species range. And the DNA delivery method also, it's very difficult. Even though I say the biologistic method is efficient, it's like a variable, species to species again. We cannot say how efficient compared to nuclear transformation, chloroplast transformation, obviously they are not that efficient, they are lesser. That's why more works are done in the nucleus, not in the chloroplast. By that itself you can understand. Still, this has to be overcome. Better method, we can say we expect a more promising method to deliver the genome into the chloroplast genome. And homoplasmy, that problem in higher plants I was mentioning, it requires multiple generations to achieve the homoplasmy. It's not like, okay, I generated, like my Chlamydomonas I can do it. One generation, single nucleus, single chloroplast, I can able to transform. With one selection, I can go for it. But in terms of crops, it has multiple genome, multiple plastome. So until you achieve the homoplasmy, we cannot go for any commercial values. So it's time consuming. And markers, the limitations of markers. Still many markers has to be developed. And in terms, there is a debate that markers should not be used. Even in the genetically modified organisms, the rice, whatever we develop, people are not ready to accept them because of the markers they use. So if you are going to increase the markers, again later when you are going for commercialization, problems may come. So there should be some alternative for marker free in this method also. And protein expression. And one of the interesting point yesterday Tripati sir was talking is about the genome. It's more prokaryotic. No? So obviously prokaryote does not have any post-translational modification, glycosylation, the foldings. So all these processes will not be there. So obviously when you go for eukaryotic products like any vaccines, those stuff, the quality will not be there, or you cannot be able to achieve what you expect. So that's what they are limiting therapeutic purity value of protein yield. So on regeneration I already mentioned, they are still struggling due to the mechanical damage. Still the better efficient method has to be developed. And containment, they are telling it's 100% maternal, but still now and then some reports are coming. There seems to be some leakage. So that has to be taken into concern whether they are 100% maternal or some exception are there or not. So that has to be still completely gone through. And genetic tool I have already said. Burden, and this is one of the important points. We are trying to make the chloroplast to work as much as possible for our requirement, not for its requirement, not for the host requirement. So obviously the metabolic burden will increase. So the investment of energy for our resources will affect the growth of the plant. So that's what's happening. So as you are going to progress in the chloroplast transformation, it is going to affect the host growth and development. So you should take care the balance, that is very important. And regulation, as I already mentioned, the selection markers, whatever you are going to use, will be a concern in the GMO. So these are all the points to be taken care before stepping into the commercial crops. That's what I need to speak to you today. That's all from my side. You can ask me anything if you have any doubts on it. Understood? You can ask me anything from this topic. Yeah. chloroplast vector means the one you designed or the available nuclear?
Like, this is possible, or the one they were using, will it have like high copy number?
No, for now they haven't tried like high copy number, low copy number. They just tried with the available commercial, because here the copy number they are not replicating inside, right? We are externally sending. The origin of replication, what we are giving, is externally we are transferring the entire genome, but there it's going to integrate into the nuclear. They are not replicating as a plasmid inside. So there the requirement for replication not necessary. Only in the E. coli where you want to multiply before transformation, this question should be answered. Inside they are going to use the host translation mechanism.
So how even those 5% cells are able to regenerate?
Yeah, this is not, yeah, obviously from my personal experience, I work on Chloroplast and I do nuclear transformation, not chloroplast transformation. So I will be taking, let's take 50 cells I will take, and I will be transforming using my own method, not this method, I use electroporation. And obviously the load you give, whatever the gene, foreign gene, whatever you give will be a negligible amount. So you transform, and not everything will be again going to be transformed. Cells, not the gene which you send, will not get transformed into the gene of interest. As you asked, only 5 to 10 will get transformed. So your question is,
How even those 5% of cells are able to?
yeah, because they have the selection markers, right? And this is going to be tissue culture based. So in tissue culture, how you will go? A single cell or protoplast, we can able to generate into the plant cells. That's the principle of the totipotency, if you are aware of that, right? So even a single cell, they have the potential to develop into the entire plant cells. So the advantage of the selection marker is they can able to confer resistance to the antibiotics. So even one cell has the transformation with the selection marker, they can be able to resist the antibiotics. Once they can able to resist the antibiotic, they can able to use the totipotency and they can live. That's the only advantage in the plant cell. If you go through animal cell, it's difficult. De-differentiation, re-differentiation, these things are all you might be knowing, right?
Sir, electroporation we cannot use for chloroplast, right?
Chloroplast transformation, it's not efficient method. I read about this because I use chloroplast for nuclear transformation using electroporation. But for chloroplast transformation, they are suggesting it's not ideal. They use PEG as well as the biogenetic method.
There will be a sir detected any transformation, accident transformation to chloroplast during nuclear transformation.
There should be, I haven't checked it. Obviously, we go for the selection marker, which are specific to the nuclear antibiotic genes. The promoters, one thing, the promoters you use should be specific to the chloroplast. Then only whatever transformed into the chloroplast will express. But whatever the construct I made will be expressed in the nucleus only. So in that case, there may be chance, as you said, out of 100 cells, one might accidentally got introduced. But in the selection process, I would have neglected them. Because even though they are transcribed inside, they are integrated, the promoters will not be active there. So they cannot able to express the antibiotics. So in that cell, they cannot able to resist the antibiotics. So in the selection, I would have eliminated. But it's very inefficient to use the electroporation.
chlorpridine, and you can see the about to red, right? Right. Then skip. CH3 becomes CHO, right? Group A to B, or binary group becomes CHO, right? And group AD. Okay? It's a big shift, right? In the adjustment property. Big change. Why this? Huh? No. Not oxygen content. If the oxygen is had electronegative, you know, what happens? It pulls the electron. Okay. Alright? So it pulls the electron from the pi ring of the apparatus. What the tetrapy of pi electrons, right? And what it pulls it, so there are certain changes in electron structure of the molecule. So that changes the absorption spectrum. As I told you last time, if you know the electron structure of the molecule, you can, and in the Boole quantum mechanics, you can calculate the absorption spectrum. Okay? So you don't have a visual. Okay? So it's all reflection of how what is electron structure. Absorption also fluorescence. Fluorescence also the same. Okay? Fluorescence spectrum is also determined by the atomic structure of the molecule. The quantum field of this fluorescence is also determined by the electron structure of the molecule. And that may change in environment. Quantum field of chlorophyll A in 80% acetone is not the same as the quantum field of chlorophyll A in a leaf. Okay? Much lower than in chlorophyll A in transport. Okay? Because absorption fluorescence property decreases. If you see tryptophan or in a protein, tryptophan or protein, okay, its absorption properties quantum field also changes. Quantum is quite high for tryptophan in around 0.3, tyrosine around 0.2 or 0.28 or 0.3. But if you put the same tyrosine in a protein, it quantum drops 0.3, 0.03 or 0.05. Around 10, 90% drop in the quantum field. Okay? So all is so you see most of the proteins will absorb in 280. Fluorescent to where? 350 protein. Why does that so good? Mostly tryptophan, right? Tyrosine, why does it absorb? Which are they? Around 270, up to 7. And that fluorescence around 310 nanometer, up to 30 nanometer. Okay. Anyway, so that is because the fluorescence is mostly at 350. Although there is tyrosine, the quantum of tyrosine dropped very severely when it bound to protein. Okay. You can see tyrosine fluorescence if you can either optically silence tryptophan by modifying tryptophan, like tetranitromethane or something, they modify tyrosine or tryptophan, you can see that. Maybe NATA will modify tryptophan. Okay, optical silencing. Or there are many other means also you can see. Can you see can be a protein which does lack tryptophan? Proteins that lack tryptophan, no tryptophan. plastocyle, doesn't contain any only carotene. Okay, calmogene, okay, insulin, okay, they are the proteins which do not contain any tryptophan. So they will fluoresce around 310 nanometers or 312, 13 nanometers. Okay, and they are observed also very low, 267 up to 80, 60, 80 nanometers. Okay, okay. Let's putting something else here. Okay, go ahead. Okay. Okay. Now transition from unicellularity to multicellularity, right? Okay. Two. All right. Oh, sorry. Most of these students are here, sitting here. So I thought I teach all of them. Transition from unicellular to multicellular. What is this? Okay. So that unicellular life, okay, eukaryotic life, what? 150 billions of years ago, okay, eukaryotic. Then, after some time, in process evolution, of course, a chlamydomonas-like organism developed. And then you can see a loose association of chlamydomonas-like organisms to form a loose association. Okay. As a forward, as a transition between unicellular to multicellular. Okay. You can see here Pandorina, Pandorina, Nidorina, Gonium, Idorina. There are the transition forms from chlamydomonas. These are loose association of what? Unicellular cells. Okay. To a so-called multicellular cell, valves, okay, which have cytoplasmic bridge between them. Okay, cytoplasmic bridge, right? Each cell is connected by cytoplasmic bridge in valves, right? So if you see the valves, the cytoplasmic bridge is there between the cells. Not only that, it's a multicellular organism which is there. Because when it moves, a certain group of flagella move like this in front, and just like and a certain group of flagella are behind, will go this way. Okay. So the one is how they go opposite direction. The front in one way and back in another way. So you propel from one to another by movement, right? The coordination between the cells. Not only that, there is a differentiation. You can see the valves. Okay, these are reproductive cells. Reproductive cells inside the valves called vegetative cell. The clear differentiation. Vegetative cell and the reproductive cells. Okay. Specialization. Gonidia, 32 cell embryo. These are 32 cell embryo. Okay, gonidia. Asymmetrical cell division, large cells, go to reproductive cell, small somatic cell. Okay. So instead of equal division, they undergo asymmetry. Okay. The smaller one becomes vegetative cell, bigger one it becomes the reproductive cell. Okay. Alright. So in small cells, reg A gene is on. So therefore somatic cells are replaced. Somatic cells replaced. Last cell, lag is on. So reproductive cell, somatic cells. Okay. So somatic cells are replaced. So it becomes a somatic genes. Somatic genes are somatic, you understand that, right? Vegetative. Okay. somatic genes are suppressed, so in reproductive phase. Here, the gonadal genes are repressed, so it becomes the vegetative phase. Okay. So you can see a spatial separation up to different kinds of cells. Vegetative cells in one place, reproductive cells come going into different place in the colony. Okay? But in the Chlamydomonas, of course, you also reproductive cells and a reproductive phase and a vegetative phase. But what is happening here? A temporal spatial is temporal separation, time. Okay? The spatial separation is a temporal separation, Chlamydomonas. Right? What happens? Rega is on somatic cell or bioplastic is pressed. Lag on, cell lag is on, reproductive cells or cell pressed. Reproductive cell on the control cell, somatic cell. So at a certain phase of time, the chlamydomonas cells are what? Chlamydomonas cells are what? Vegetative. Sometimes they themselves become reproductive. Okay? So it is temporal separation of vegetative phase and the reproductive phase in chlamydomonas cells. But a multicellular organism like volvulus, okay, where there clear connection between two cells, intercellular connection by cytoplasmic bridge, there is a separation of vegetative cells and gonadal cells, but they are all taking place at different places of the cell. So this is a colony now, right? Okay. Now, in the process of evolution, this colony became replaced, compact. Okay? And you got a system where you have now the two cells are in contact. Okay? So you have plasmodesmata, connecting same cytoplasmic bridge exists in a plant, right? Two cells are connected by plasmodesmata. You know plasmodesmata, right? The connection bridge between two cells through the cell wall, right? Right? So the plasmodesmata connects two cells, right? So there is no movement of nutrients or signals or hormones. Everything goes by a plasmodesmata from cell to cell. Here it goes from cell to cell by cytoplasmic bridge in the volvulus. Okay. So unicellular to multicellularity also led to adhesion of the adhesion between two cells, connected by cytoplasmic bridge called plasmodesmata in plants. And in animal cells, there is the same thing took place, but they are contact cells. Two cells in animals are connected by contact cells. We call them contact cells in animal cells. Okay. All right. In our body, two cells are connected, so called contact cells, where there is also a bridge, okay, between the two cells. Okay. So these intermediate form is a colonial organism. Okay, they are only made a colony. Okay, but they are not connected by intercellular intercytoplasmic bridges. These four examples. Okay, then that slowly developed into a colony-like multicellular organism like volvulus, and they are connected by intercellular organisms. You can see the volvulus. One here in many colonies of volvulus, right? In a single, okay? Many colonies. Okay. Now the question arises. So chlorophyll is green. Why chlorophyll is green? Why is the chlorophyll green? Huh? Huh? Because it is observing green light, right? All the colors they observe all the wavelengths of light other than green, right? You can see here. Here, right? None of the observing green, you see. I told you that, right? But the green, 500% here. They are not observing anything, right? So it's a big stretch of wavelengths where they are not observing that. Okay. Don't you think ideally chlorophyll should have been black? Ideally. Plants should have been black. Other than green. Why? Because they should observe all wavelengths of light. Right? They are black. When they black, a black plant, a black spot, or black color, any black color, observe all the wavelengths of light. Right? So plants should be ideally black, right? To harness all the wavelengths of light and convert it into food. Right? They can carry energy. But they haven't made their green. Why do you think so? Why there are big deficiencies? There is no direct answer to this. Okay, I will give you some explanation. Okay. I don't have the direct proof what I am saying. Okay. Alright, I am going to say now. Then because we are talking about evolution here, okay? Evolution doesn't have a direct proof, okay? Okay. You know, anaerobic world, okay, here. We don't have any black chlorophyll yet. Okay. There may be some other pigments associated in the plant, in the organism which look like black but is not really black. Okay. Some very, very old cyanobacteria organisms would look black but they are not really black. They cannot absorb green light. Okay. Okay, I am just going back and come back to you later. Yeah. So this is anaerobic world, right? No oxygen, right? Unoxygenic world, right? Okay. And what prevailed? Archaea, right? As I told you, archaea prevailed here. Archaea is primitive organisms, right? Very, very primitive bacteria. They evolved here. And have you heard of a bacteria called Halobacterium halobium? Halobacterium halobium. Its old name, the old name was. Its now name is changed now, but it's still known as Halobacterium halobium. There are many bacteria like that. Millions of them. Okay. Thousands of them. What did they do? They don't have chlorophylls. They don't have bacterial chlorophylls either. They have but they are autotrophic bacteria. Or they can synthesize their food of their own. Okay. What to do? They have rhodopsin, bacteriorhodopsin. You know, when you go to nature, we feel very good, right? Why? Huh? Huh? Fresh air? Okay. Any other reason? Nature is green for us. Nature is green. Okay. And we are very nice at it because what is this? Our rhodopsin neurons focused on green light. Okay. Our ability to see the green light is maximum. Rhodopsin absorbs maximally in the green. Okay. And our ability to see the blue light or red light drops drastically. Okay. All right. Very, very low. Okay. So with the least energy, we can see green light. Okay. So we are so comfortable in green light or looking at anything green. Okay. All right. Okay. Similarly, Halobacterium halobium, as I told you, has a bacteriorhodopsin. We have inherited rhodopsin, our rhodopsin from bacteria, anaerobic world. Okay. Rhodopsin that prevailed in billions of years ago, around 3.5 billions of years ago. Okay. We have inherited bacteriorhodopsin and converted to our rhodopsin. Okay. Slight change, not much change. Okay, in bacteriorhodopsin and our rhodopsin and I, same, almost same. Structure is almost similar. Okay, we have selected nature, you know. Okay, so because that is a pigment that was selected for animal eye. Okay, okay, invertebrate eye, vertebrate eyes. Okay, all right, invertebrate pigment, vertebrate pigments. Essential landing up in human eye. Okay, evolutionary. Okay, all conserved. Okay, and if you see in anaerobic world, archaea prevailed, right? So it's full of archaea. Okay, and photosynthetic bacteria or cyanobacteria developed below archaea in the ocean, right? What? Okay. If you see the red one, red one, okay, that is the absorption spectrum of bacteriorhodopsin. Bacteriorhodopsin. Okay. And this is bacteriorhodopsin, structure of bacteriorhodopsin. Okay. Helix. It also retinol. Okay. Vitamin A, right? Okay. So retinal linked to lysine. So this hydrophobic core of bilateral lipid membrane. These are spanning the membrane. Okay, this is structure. And so the N-terminal end, C-terminal end, in the hydrophobic core, we have this retinol. Therefore, it's same thing. Same vitamin A is also in our eye, retinol. Okay? Okay? That's the chromophore, right? You see the retinol. Retinol. This structure. That absorbs the pigment, green light. Okay? Therefore, this is green. That is the absorption spectrum of that retinol. Okay? All right? And this is in the membrane, lot of retinol attached here. This is a structure of bacteriorhodopsin, trimeric structure of bacteriorhodopsin. It is trimer. Okay? And you can see retinols here. These are all retinols here, right? Retinol, right? You see 3 retinols in the structure. Okay. So they are all responsible for absorbing green light. Okay. And there is no difference between structure of bacteriorhodopsin
and human rhodopsin. Same trimeric structure. Okay, say exactly almost same. Okay. Slight difference in the protein structure. Retinol remains same. Okay. So you say evolution conservation, not from, it's from archaea to human beings. Right? Billions of years. But see the conservation of nature. Okay. Not evolved. Okay. New pigments. Okay, conserved. Okay. Okay. So now when I say the earth is full of what? Archaea, which contain what? Rhodopsin, bacteriorhodopsin, that absorbs a wavelength like this, right? As I said, it is also focused on green, 540 nanometers. Where the peak? 540 nanometers, right? And it is absorption decreasing to 700 here and decreasing to 400 there. Okay? All right. So that's retinol, right? That observing there. And when these cyanobacteria, kind-like organisms, or even bacteria, photosynthetic bacteria, they evolved below the archaea, they did not, what all the light coming to the falling on the ocean, okay, all right, were absorbed by what? Archaea. Okay. What archaea could not observe, okay, collected below, okay, where what? This photosynthetic bacteria evolved or cyanobacteria evolved. Okay. So when cyanobacteria or photosynthetic bacteria evolved, they did not see the green light. Because when they evolved, they were green deficient in green light. Okay, because all the green light is absorbed by archaea, by retinol. Okay. So they evolved a pigment called chlorophyll or bacterial chlorophyll, which does not absorb green light. Okay. And therefore the evolution of selection of the pigment that ultimately resulted in synthesis of chlorophylls in the plants. Okay. And they did lot of permutations, combinations in the process of evolution, and finally came up with a pigment called bacterial chlorophyll. Okay. And this bacterial chlorophyll did a fantastic job for them, okay, to absorb light and convert to chemical energy, although anaerobically, right, from hydrogen sulfide, acetate, right, they convert chemical energy, photosynthetic bacteria. Okay. Then these photosynthetic bacteria, okay, through the course of evolution, simply changed their structure, shifted the wavelength from 780 to 700, then 663 to chlorophyll A, 760 to 660, save 700 for chlorophyll D. Actually, if you put chlorophyll F, 770, 700, 720 for chlorophyll F, to 700 for chlorophyll D, okay, to 663 for chlorophyll A, or blue shift, shifting to smaller wavelengths. Okay, but still they did not absorb green light, because cyanobacteria also evolved around a little later. Okay, after the photosynthetic bacteria evolved. Okay, we believe that photosynthetic bacteria is the progenitor of cyanobacteria. Okay, photosynthetic bacteria, right, which are bacterial chlorophyll A and B, are bacterial chlorophyll A is progenitor of cyanobacteria. Okay, cyanobacteria evolved from photosynthetic bacteria. Okay, all right. So they simply did some modification of side chains and shifted their wavelengths to smaller wavelengths, so they can absorb visible light, okay, and they have enough energy now to oxidize water to oxygen, okay. Earlier they did not have enough energy to oxidize water to oxygen, okay, because they are absorbing at very longer wavelengths, okay. So this conversion, okay, so shifting to smaller wavelengths, it has nothing, nowhere near green, okay, because to observe green they select a completely different pigment called retinol, okay. And retinol was not selected therefore for plants, okay. Probably they did not see retinol, green light, so they did not take retinol. If they had added retinol along with chlorophylls those days, okay, probably you have black bacteria, because they are absorbing all of light and also absorbing green. If you see the structure, if you think of organism which have this and absorption here like this, okay, from here to here to here to here to here, okay, all wavelengths of light absorbed, right? So chlorophyll would have been black, right? But it was not black. Why it was black? It's because they absorb all the wavelengths light. Okay. So and then once chlorophyll is selected, okay, from bacterial chlorophyll, actually, okay, the structure is microcycle has no change, only the peripheral, as I showed you, there is only change in the peripheral side chains, right? I told you that yesterday, right? Only side chains have changed, side groups have changed, no change in the macrocycle of the chlorophylls through the billions of years of evolution, right? So the structure, therefore they could not absorb the green light. They changed, could not. Okay, they came out some other pigment like carotenoids and all that, but carotenoids doesn't make them black. Carotenoid doesn't make them black. They absorb 470 nanometers maximally. Okay, carotenoids. Okay, so that's okay. Okay, and plants got used to for the green light out. Okay, and therefore they you see, even the observed energy we plant has with this mostly blue and red, right? The major two wavelengths where absorption takes place, blue and red. Okay, this blue and red absorption is sufficient for the plant actually. Okay, because it has so much energy now. Okay, it produces NADPH, right? Reductant is producing. Okay, then limitation is Rubisco. Limitation is Rubisco. Okay, always Rubisco is a limitation. Okay, because Rubisco is a sluggish enzyme. The turnover rate is very low. Okay, very low. Can you give a very fast enzyme turnover rate? When I say Rubisco is very small enzyme, sluggish enzyme, I don't have written here. I don't think I have written here in the slide. It is not here. It is not here. Yeah, this is something else about the slide. No. This is slide doesn't work. There are other slides I can show you, but there is no need for me to show that. So there is no need for me to show that. Okay. Carbonic anhydrase is a very fast enzyme, high turnover. What is turnover rate of an enzyme? Yes, binds to substrate, converts the product. Once convert product released from the enzyme active site, they again ready to bind to a substrate, right? Again form the product released from active site, again bind to the substrate. So how many times it can bind to the substrate forming product. So how many times it can bind the substrate to form the product, okay, per second, determines the turnover rate of the enzyme. Okay, all right. So, okay, so when a sluggish enzyme means turnover is low, means it cannot form the product. It can very fast, Rubisco. Okay, it produce slow. Carbonic anhydrase is another enzyme which is very fast. Carbon dioxide plus what carbonic anhydrase does? CO2 plus water from bicarbonate, right? Bicarbonate. That's the product of carbonic anhydrase, right? And that is very fast reaction, very, very fast reaction. We called it, if we call Rubisco a rabbit, Rubisco is a, what is not rabbit, what is the race was between, not crocodile. Yeah, that mollusk, what is that? Turtle, right? Rubisco is a turtle which moves very slow, right? Carbonic anhydrase is a vehicle moves very fast, is like a rabbit, right? Runs fast. Okay. All right. All right. So this is our, okay. So what I said you, CO2 concentration in atmosphere is increasing, right? CO2 concentration in atmosphere is increasing. And temperature is also increasing. When CO2 concentration is increasing, what is going to happen? What is going to happen to the plant productivity? And plant productivity is going to increase. Why? Because photorespiration is suppressed. Because photorespiration is suppressed. Okay. The Rubisco, there is always a competition between oxygen and carbon dioxide. Okay. All right. So suppressed. But this increase not always. I've written down somewhere here for you. Not always true. There is also down regulation of increase in carbon dioxide concentration, okay, to result in stimulation in photosynthetic carbon assimilation fixation between 30 to 50%. Okay. Primary due to reduction is photorespiration. Okay. Either the Rubisco carboxyl reaction favored in this condition. Oxygenation is suppressed. Okay. But it's not so. However, many plant species, grow elements you do not have, engaged photosynthesis goes up 30 to 50%. Okay. Why do you think so? Why that happens? When you have more carbon dioxide in the atmosphere, okay, and less of oxygenation, more of carboxylation, then plant thinks, why should I invest so much my energy? Okay, one protein called Rubisco. Okay, I can diversify my amino acids to some other useful proteins. Okay, do you understand that? The plant therefore reduces Rubisco content in its own body, in the leaf. So Rubisco concentration very often, not always, but many times, many species are reduced in high CO2 environment. Okay, if the Rubisco concentration decreased, then you will not get a very increase in photosynthesis. Even if oxygenation is suppressed and carboxylation is favored. Okay, in high CO2 environment because Rubisco is suppressed. Its substance is less than this figure. This is probably because the plants are able to exhibit acclimated down regulation, decreasing photosynthetic potential, particularly long-term growth related CO2. This anaerobic active response is often correlated with increased carbohydrate levels to get the reduction in total nitrogen Rubisco activity. Okay, nitrogen content decreases, protein content decreases. Okay, so it is not only even high CO2, high CO2 in climate change, high CO2. Okay, although we have more carbohydrates in this site in the plants. Do you understand that? More carbohydrates synthesized. So for, let's say, per grain, we had 1 grain, let's say, weighs 20 grams or 100 grams. 1 grain, let's say, weighs 10 grams. Okay, 10 grams of one grain, let's say, has 1 gram protein. So 10% is the protein. Okay, all right, maximum in a cereal. Okay, all right. Now, when plant assimilates more of carbon dioxide, okay, manufacture more and more carbohydrates, right? More and more carbohydrates. So the what is nitrogen? Where comes proteins come nitrogen? Where nitrogen comes from the soil in form of nitrate, right? So nitrate assimilation, nitrate reduction to ammonium, and subsequent assimilation of ammonium to amino acids. Okay, okay, cannot keep pace with the increased CO2 concentration fixation. Increased CO2 fixation. CO2 fixation is high, the carboxylation is high. Okay, but so we have more carbohydrate, but you cannot synthesize amino acids and amino acid proteins at faster rate because of limitation nitrogen assimilation. Okay, nitrogen content in soil not increasing. What is increasing? The CO2 atmosphere. You may have some, but if you increase the nitrate input into the soil, okay, increase the fertilization level in the soil, probably you will not have a reduced protein content. Okay, but in natural ecosystem, okay, natural ecosystem, the tree or a plant or any other plant, okay, you have carbohydrate, you have in a seed you have more carbohydrate but less protein. You understand that? Huh? Because amino acid content, nitrogen assimilation will be low. Okay, nitrogen assimilation means same, it is not going to be reduced. Nitrogen assimilation will be same. So protein synthesis will be same, but there will be more carbohydrates because of CO2 concentration atmosphere is increasing. So you have more carbohydrates and less protein in the seed. So wheat plant growing in nature will have more carbohydrates and less protein. They are not the carbohydrate seeds in elevated CO2. So this may lead to protein deficiency in the plants. In the seeds, of course, we may face a protein deficiency later on. Okay, but all these things are good enough if it's not accompanied by high temperature. But as I told you before, high CO, and you know by now, high CO2 is going to accompany by high temperature. Okay, then what is going to happen? Tell me, what is going to happen? When high CO2 and high temperature, what is going to happen? Think and tell. Huh? Huh? Enzyme? Enzyme? We are not going to denaturing phase. Enzyme denaturation is very in a high, very high temperature. Okay. Huh? that is viscoactivity and all that will be affected high temperature. But that is a different story altogether. Reproductive growth will higher, the vegetative growth will higher. Okay, you grow at high temperature, little bit high temp, 1 or 2 degrees centigrade increase. We're not going to have 10 degree increase in temperature. Okay, we will have a 2 to 3 degree, maximum 3 degree increase in temperature. That will may favor crop growth. Okay, but it will reduce the reproductive growth. Pollen grain setting will be reduced. Pollen grain germination will be reduced. Pollen grain formation will be reduced. Okay, when pollen grain formation is reduced, what is going to happen? Fertilization will be less. Okay, flowering fertilization less, so less seed formation. So you will reach a scenario where you have more vegetative growth and less reproductive growth. So what is going to happen? You have more vegetative mass, but less seed formation. So it is going to impact our agriculture if high CO2 is accompanied by increased high temperature. You see, 2-3 years ago, in month of February, the temperature went to 30 degrees in northern India, northern belt, but a lot of wheats are grown there. Wheat is grown there in northern areas, in Punjab, Haryana, all that places, Uttar Pradesh, and all that. Okay, they grow wheat, and wheat flowering time is in February, January, February. February temperature increased by 4 to 5 degrees centigrade. Okay, average temperature of February increased 3 years ago, 25 degrees centigrade. So that led to reduction in pollen grain formation, reproductive decrease, and reduce seed production of wheat. Okay, and therefore seed production was less. Okay, because pollen grain didn't germinate, pollen grain was not formed or did not germinate on the stigma. Okay, all right, no fertilization, therefore no seed formation. Okay, so it's highly reduced, highly suppressed. So even 2 to 3 degree increase in temperature may not create a big problem for vegetative growth of the crop, but may affect the seed setting, flowering, and pollen formation. So we have more biomass and less seeds. Okay, so the advantages of high CO2 for favoring more vegetative growth will be gone at high temperature, especially for wheat, even for rice. Same thing, 3 degrees centigrade is more than enough, average increase 3 degrees centigrade. It also will decrease the reproductive growth in rice also. Okay, and this is going to happen. Okay, because we've already seen increase in temperature. Okay, right? temperature बढ़ा है कि नहीं है? बढ़ा है ना? How much? कब बढ़ा है? कितना बढ़ा है? I don't know, I have that slide with me. Here it is. Here you see Arctic region. What is Arctic? Northern pole. North pole is Arctic, right? The temperature measurement Arctic. This is measured by NASA, 1880, and I have figured it 2004, but it is 25, is much higher. You can see the temperature constantly increasing here, right? This is minus. If this is today's 0, the minus 1.5. Okay, we are giving that to crossing 0, going up and up and up here. I'm sure you've gone up by now. Okay. So Arctic has already experienced around 3 degree increase temperature. Arctic region, not of the whole world, but the North Pole. North Pole has already increased its temperature last 120 years. Okay, one to 3 degree increase temperature. Okay, therefore glaciers are melting, and all that. Why do you think Arctic is increasing more than Antarctica? Antarctica temperature is less increase. Arctic is more, North Pole. More of human activity there, more human activity in Baltic, Northern Pole, North Poles, than South Poles? No? What? Hmm? Yeah, ozone layer depletion is there, but that may not affect much of temperature. It will? Huh? It will? Why? Why? Huh? UV radiation is more. Of course, UV radiation is more there. Okay. Huh? Arctic black. Arctic, there are a phenomenon called albedo. Albedo. What is albedo? They are all ice, right? All ice, right? And when light is falling, also going up immediately. Okay, okay. Light is striking, and in the ice, reflecting back. Okay. So if it reflect back to the atmosphere, it's fine. Okay, but it's observed on Earth, then temperature is going to increase the temperature. Okay, it goes back fine. Okay, so this if we have green leaves there, plants there, it will have positive effect there. Okay, you observe light and increase the temperature. Leaves there, we're going to observe the light and increase the temperature. Do you understand that? So we don't want in North Pole region, polar region, where albedo is working for us. Okay, it's coming and going. Okay, not being observed by Earth. Okay, okay, getting reflected back to atmosphere. Okay, if that is changing, okay, okay, it is reduced. Reflection is reduced by absorption of green plants. Even small amount of green algae that is growing in the Arctic ice would change the albedo, change the reflectance. That will increase the temperature. Okay, that is one of the impact. Okay. Okay. So, do you understand what I am telling about this aspect? I don't know. The recruitment of these 3 proteins, you know about this? Oxygen evolving complex, right? You know oxygen evolving complex. What it does? I think he must have taught you by now, your Japanese collaborator. Did you talk about anything about that? Oxygen evolution or oxygen uptake? No. Okay. Takahashi, na? Okay. What he taught you? LSC. Huh? Structural biology. Oh, yeah. This is also part of structural biology. We know about manganese for complex structure and all. Okay. I'm sure we'll talk about it, so I'm not going to talk about it. So I have left it for him. Okay. Now, another point I must say, yes. So this is how to combat elevated CO2. Okay. Reduction of greenhouse gas emission, of course, very important. CO2 emission in particular, and then methane, right? Methane emission. Methane emitting today very minimal, very low, but still some amount of methane is emitted from the rice fields. Why? Why? Because water, rice needs a lot of water. Rice grows in green rainy season, okay, and the earth is full of water, water logging. So there is no oxygen on earth. The air occupied, the space occupied by air is now filled by water. Okay, so there is no air. So there, what will grow? Which bacteria grow there? Anaerobic bacteria, right? So anaerobic bacteria will grow in the rice field. Okay, in anaerobic conditions. So they will emit methane. Okay, right? So the methane emission also causes global warming. Okay. So you have reduced the greenhouse gas emission. Increase CO2 for plant, especially reforestation. You have cut down so much, 70% of the forests. Okay, cover. Can you restore it back? It's a very important problem. As I told you, you restore it, we will certainly decrease temperature. Certainly. There is no doubt about it. Okay. You plant trees, enormous number of trees, all around where you're staying, around the globe, wherever you can. Okay. Will certainly reduce the temperature. Certainly. I told you reason why, right? Because I told you the plants transpire and latent heat evaporation is provided by the leaves, and therefore it cools the atmosphere. Okay. Plantation of fast-growing trees is very important, like Populus deltoides or some other, or many other species. But today, Populus deltoides may not be a great choice, but any tree is fine, may not be fast-growing, but they should be strong enough. Because, you may experience this, but coastal India experience always what? The many hurricanes, right? Right, hurricanes. And these come and destroy, devastate their trees. Okay. And that is mostly due to the fact that there are, many plants, coastal plants, mangroves. Okay. Mangroves were there everywhere throughout the, if you cut from Bengal to Cape Comorin, Kanyakumari, back to Gujarat coast, throughout. Okay, we had mangroves. Okay, you have destroyed mangroves like anything. Very few pockets in India have now mangroves. One probably in Sundarban, another maybe Bhitarkanika in Odisha, and another maybe Pichavaram in Andhra Pradesh. Huh? Kerala also have some mangroves. Some mangroves. That's it. Goa may not have it also. There may be some mangroves in Goa also, but not much. Okay. So if you three few localities, not mangroves in Kerala, mangroves not everywhere. In some places. That's what I'm saying. Not everywhere. This is the mangrove forest. Do you see this? of Sundarban. Okay, anybody have gone to Sundarban? These dense forests you not see anywhere else. Vithakani mangroves are there, but not that dense. It's there, not that dense. Okay, Sundarban is full of mangroves. Okay, Vithakani mangrove, but a small place. And Sundarban is very big, is a big, vast place. Okay. So, what we are doing, look at the roots. Look at their roots, how they are holding the soil. And when air comes, hurricane comes, you know, they remain as wind barriers. Here, Sundarban has a lot of mangrove destroyed. Okay, they used a huge wind barrier, 4-5 kilometers long width, wind barrier. Okay, so they could reduce the intensity of hurricanes. Okay, intensity of air, speed of air. Okay, and when air reaches the landmass, okay, its speed was reduced. Therefore, it was not impacted much. Okay, and what is happening now? They are all gone. So we are all subjected. All coastal belt is subjected to very high speed of wind. Okay, and that is destroying, uprooting all the plants, all the trees. Most of the trees are gone. Okay, the big trees are gone in hurricanes. Okay, so that is one of the reasons why there is also global warming. When coastal belt is so cool, now it's very warm. You people are from Utkal University, right? So it is very warm there, very hot there now. You're not there when we are studying there. Even we are studying there in Bhubaneswar in 70s. It will be hot for a while, for 2-3 hours during daytime. Then evening, it all, even afternoon at 4 p.m., it will so windy and all that. Cool, hot is gone, and nights are very good, pleasant. Evenings are very pleasant. No problem. Only 2 to 3 hours. It's from 10 to maybe 4, 11 to 4, 4 to 5 hours. Then it's gone. But now all the time I have hot air in Bhubaneswar. That is because it is full of concrete, number one. Got all the forest gone from Bhubaneswar. All the forest, and the 99 cyclone came. That was destroyed all the forest, big trees, okay, from everywhere. 99 cyclone, and followed by many cyclones, okay. And they have all destroyed the forest cover, and whatever forest remained, man-made invasion of cutting down the forest and building back buildings back into concrete and change the temperature. Okay. See, Earth has a big light absorbing power. Okay. But concrete doesn't have that much power. Okay. The infrared light, it absorbs very deeply, and it produces heat. Infrared light, it produces heat, na? This is another component, so reflection in the urbans. These are respiratory roots. Do you see the respiratory roots? Huh? Nematophores. Yeah, coming from the soil. They are respiring in the salt habitat through the nematophores. They are roots. Roots need oxygen, na? So roots are respiring through the nematophores. Okay, because they don't have oxygen in the soil, all occupied by water. You see the root system, conserving soil, trying to conserve soil. Okay. can we come to an alternate fuel like hydrogen, right? When it's a green fuel, alcohol is not a green fuel. We are trying to mix alcohol with petrol, right? Petrol with alcohol, right? To reduce the consumption of petrol, doesn't matter. But still emit alcohol also burn to carbon dioxide. Okay, so it's not a really green fuel. It's also producing carbon dioxide. We're painting green, I don't know why. Okay, all right. One reason being that it is coming, but here the CO2 is recycled. CO2 is absorbed by plants, right? You produce alcohol from the plants, then alcohol is consumed again. So same carbon dioxide is sent back to atmosphere. Okay, so it doesn't lead to reduction in CO2 concentration from the atmosphere. So and in a country like India, how much grain we can destroy? To make 20% alcohol, blend 20% alcohol with petrol. Okay, probably we have to destroy 2 states like Kerala. Okay, 2 states area-wise. Okay, grow all crop. Okay, and that crop you are producing alcohol from there. Okay, area-wise. Okay, then only you can probably meet the demand of alcohol. Okay, to be blended with what? Petrol. So that is not a feasible program, sustainable program. Okay, not only that, you are destroying the land mass for what? You are using nitrogen from the soil, all the nutrients from the soil, and doing use to burn it as in the fuel to carbon dioxide. So the amount of nutrients you need for your survival as food is being depleted from the soil. Isn't it? So you are having more and more fertilizers, and they're creating more and more harm to the soil. Okay. So instead of doing that, if you think of hydrogen as an alternative fuel in car, hydrogen fuel. Electric vehicles are okay, but electric energy is not coming of your own. Electric energy also coming either from thermal energy, burning coal, okay, or hydroelectricity, making vast devastation to the landmass, right? Making many people displaced, okay, when you are making water reservoirs, okay, all right. So it is, and also in Himalayas, it's creating a lot of havoc there now. All landslides are taking place in Himalayas, all country landslides, huge landslides, because you make dams, and they water it accumulates there, and the water percolates into the rocks in the mountains, and then rocks become slowly comes out. Okay. Okay, the adhesion of the rock between rock is gone. So then what happens? The rocks fall down, land mass fall, big landslides take place. Okay. So it happens. Okay. All right. So if let's say like this. Hello. Good afternoon. I am not there till tomorrow. How do you produce hydrogen biologically? Can you produce biological photosynthetically? at the expense of solar energy. Hydrogen can produce photolysis of water. Photolysis of water, but you don't know, put electricity, pass electricity in water, will convert hydrogen and oxygen. You can get hydrogen from there. A vast ocean, no problem. But energy input is quite high to hydrolyze water to oxygen, water, hydrogen, oxygen. So idea is, can you do it at the expense of solar energy? We are receiving, okay, directly from the what? Directly from water. With photolysis of water through photosynthesis, right? So the enzyme called hydrogenase in many algae. Chlamydomonas is one of them. Very important, okay, one of them. What it does, once ferredoxin is reduced ferredoxin, okay, then hydrogenase, okay, converts reduced ferredoxin to what? Oxidized ferredoxin, hydrogen. Here reduced ferredoxin does not give electron to NADP, for NADPH. Rather, it gives reduced ferredoxin gives electron to is now oxidized to hydrogen. Okay. All right, by enzyme hydrogenase. Reduced ferredoxin, you know, from photosynthetic electron transport chain. The ferredoxin by FNR, ferredoxin NADP reductase, gives electron to NADP to make NADPH. But before it gives electrons to NADP, you give what? Hydrogenase, which is already present in the plant, in the algae. Okay, so it reduced. It gives out hydrogen to you. Okay, okay, it's so simple to say. So this is what is it, right? Let's say pH2, right? Water for 4 protons, right? Now, this what happens under normal conditions, right? NADPH production and all. NADPH conversion. If you make it a little bit anaerobic atmosphere, okay, little anaerobic atmosphere, then what will happen? Let's see. Reduce ferredoxin here. Reduce ferredoxin, right? Ferredoxin works as hydrogenase to form hydrogen on anaerobic condition. Okay? Because hydrogenase survives only anaerobically. Hydrogenase enzyme is destroyed by oxygen. Remember that. Hydrogen is destroyed by oxygen. So if you can maintain a little bit anaerobic condition, okay, in the cell, okay, then you can produce hydrogen. Okay. So we are talking about this production of hydrogen, anaerobic conditions. How can do that? Yeah. So there are fuels, oxygen microbe, hydrogen fuel cells.
Excuse me, sir. Sir, excuse me, sir.
Yes.
Just wanted to sir, in case of this oxygenic photosynthetic hydrogenases, like enzymes, hydrogen producing enzymes, they are sensitive to oxygen. So, how about this anaerobic photosynthetic bacteria, which are usually grown in anaerobic photosystems?
Anaerobic photosynthesis, okay, if you can, that is a possibility.
Yeah.
But let's say you give hydrogen sulfide as the electron donor, H2S,
Yes, yes.
or something like that. It depends on what kind of bacteria you are selecting.
Yes, yes, yes.
Then it can produce hydrogen, no problem in that. Anaerobically, because they are anaerobic bacteria, photosynthetic bacteria. what the issue is that you are again giving substrate like hydrogen sulfide. It is not water, it is substrate anymore. It is hydrogen. You are manufacturing, producing. You have to give some. You to synthesize hydrogen sulfide. You have to spend some energy. Okay. It is not all prevalent in water.
Yes, sir.
Or everywhere in ocean. Okay. Water is the very cheap source of electron donors. But whether acetic acid, acetate, hydrogen sulfide, or any other substrate, then you have to provide energy for it. So energy input is very high than your output. So water, vapor system, reduction, right? This is hydrogenate, right? Okay, hydrogen production. Okay, and you can do that. How can you maintain chlamydomonas in a hydrogen low anaerobically? So what is mitochondria does? Respiration, right? It consumes oxygen, right? Making water, anyway. So what you do, grow them in sulfur starvation. Sulfur starvation. Okay. Plant, you have reduce the sulfur from the medium, from alga. We are growing alga, but don't give sulfur to it. Okay. Or very, very minimal sulfur. Okay. So under sulfur deprivation, you can see in hours, you can see the rate of photosynthesis will drop, okay, because there is no sulfur. The rate of photosynthesis is reduced, okay. Whereas respiration is not reduced drastically, okay. So at this point, here, the anaerobic environment or not, respiration is high, photosynthesis is low. Under sulfur deficient condition, you can see photosynthesis drops. Okay, respiration remains almost the same. Okay, so at this point, after a few hours of growth or sulfur deprivation, you will see rate of respiration will go high, rate of photosynthesis will be low. So this from time period from year to year, from 40 hours to 120 hours. This is the stage where there will be anaerobic growth, anaerobic medium. Cell will be anaerobic condition. Isn't it? Photosynthesis is high, respiration is photosynthesis low, respiration is high. Right? Understandable or not? Right? Oxygen production is less, oxygen consumption is more. Oxygen consumption to respiration is more, right? Photosynthesis production, oxygen through oxygen production is less. This is all anaerobic environment. So in anaerobic condition, as I told you, hydrogen can survive. Hydrogen is functions. Hydrogen function under anaerobic environment. Okay. So there is hydrogen production. So if you measure the gas volume per ml and you measure under sulfur deprivation, you can see the hydrogen gas produced on the AA top from 50 to 100 hours. Amount of hydrogen produced. And this is actually you did in your lab, and we collected the chlamydomonas cell. You grew it at sulfur deprivation conditions, and you see, and we collected hydrogen by downward displacement of water. Okay, hydrogen collected from in the top. You see that? Huh? Hydrogen collected from by downward displacement of water. Okay, so you can do it, but economic feasibility of it, that is the point. Okay, you grow under sulfur deprivation, okay, algae, okay, and put the and collect gas from here, and you see in the top there is a hydrogen gas by downward displacement of water. Okay. So there are many ways. So if now if you collect hydrogen and put it into your car, okay, what will happen? Hydrogen will be born to consume, hydrogen oxidized by oxygen to water. So you give hydrogen, so what the byproduct is water. Hydrogen peroxide will become water. No carbon dioxide. Right? Hydrogen is the fuel driving your car. Okay. That possibility is there. It can be done, achievable target. But you need a lot of input for it. Okay. This is one way you can do it. There are many other ways you can also do it. Okay. But the hydrogen is the problem, the hydrogen is sensitive to oxygen. If you can genetically engineer hydrogenase, okay, which is insensitive to oxygen, okay, then no problem. So you knock out the hydrogen from Cambria monas and put genetically engineered hydrogenase back into the same system by transformation again on any promoter, 30 base promoter, whatever. Then it will be, you have now hydrogen, the algae does not have any original hydrogenase, that is gone, the gene is gone, and supplemented with what? A hydrogen resistant hydrogenase, oxygen resistant hydrogenase. You see, this is not hydrogen, not only enzyme. Can you see any other enzyme which is resistant, which is sensitive to oxygen? Any other enzyme? Well, nitrogen is one of them, yes. Nitrogen is also needs, is sensitive to oxygen. So, if you see nitrogen production, nitrogen fixation, what they do? Nitrogen, what they do? Convert nitrogen to ammonium, right? Ammonia, right? Convert nitrogen to ammonia, nitrogenase. And this nitrogenase is inactivated by oxygen. Therefore, you see, if you see cyanobacteria, they have heterocysts, right? Heterocyst deficient photosystem too, so there is no oxygen evolution, okay? No photosynthesis, no oxygen evolution, and a very thick wall, so that the atmospheric oxygen doesn't enter there, and whatever oxygen there is consumed by its own respiratory system. So you become a real anaerobic environment, heterocyst, where nitrogen is converted to ammonia by the enzyme nitrogenase. Read it. So in nature such system exists. So leg hemoglobin also, leg hemoglobin also fixes some nitrogen in the where? The nodules, root nodules, mediated by rhizobacteria. Not it. They have also, but they create an anaerobic environment there to fix nitrogen to ammonia. So you need, for hydrogen function, nitrogen function, you need anaerobicity. Okay. If you create a condition of anaerobicity, okay, or you can genetically engineer hydrogen, okay, where it is insensitive to oxygen. It's extremely difficult to do it, but it can be done, I guess. Okay, then no problem. You can produce hydrogen from water without fear. You know, some of them will get consumed to, it may happen that some of them will go to produce hydrogen, some of the electron protons may go to fix NADP, NADPH, carbon CO2, CO2 fixes. You never know. Okay. All right, to stop here. Okay.
want to come tomorrow.
Anna, can it's okay. Once again, I welcome you all. Guys, any problem there? Yeah, we can start the program. You can't?
That's not.
Yeah, then come half of the can come this side because camera is this side, so I cannot come there. You can come this side. Anyway, you can see the slides there, no? You can come this side. Half of the people can. Yeah. What's his name? Few of them can come this side. Move forward. Yeah. Yeah. You can still see from here. Don't worry. Come, Monica and what's his name, brother? Yeah. Come this way. Anyway, experiment part will be done here only. Just I'm showing the slides. So today, what is the work plan? Okay. So majorly, we are trying to transform the nucleus to the nucleus. Morning we talked about chloroplast transformation, no? But we are not doing exactly the chloroplast transformation. We are doing nuclear transformation. But the method will be almost similar. The instrument which you use will vary. Here we are going to use electroporator. Since due to the availability, we are using electroporator. Majorly, I have planned everything like cloning, transformation. Cloning will be a big step actually. And in order, it will be a big process. So I will be telling you theoretically how to be done. Because doing experimentally, it's nothing. Because it's like a digestion, you are going to add. Everything going to happen invisibly. So by doing, you are not going to see much. But I will be explaining you how to be done. That part I will be taking care. But experimental, you will be starting transformation. You need to imagine a way that plasmid which we are going to give contains your gene of interest. From then onwards, you will be going ahead. You will be transferring the vector, transformed vector into the DH5 alpha. Why we are doing that?
That's competent.
Yeah, competent cells. DH5 alpha is a competent cells. What is the importance of DH5 alpha? Why we are using DH5 alpha cells? More copies? No, not exactly more copies alone. There is one important point, genetic, I can say advantage. No, not high replication. What is the difference between wild type E. coli and DH5 alpha E. coli? No.
Region is there. Multiple cloning sites.
Multiple cloning sites? No. It's multiple cloning sites comes in the vector. I am talking about the organism. The E. coli, which we use is a DH5 alpha that has some mutations. So what is the feature of DH5 alpha? Why specifically we use this DH5 alpha for the transformation? That's my question. So I will be giving you the answer. DH5 alpha contain mutation in the recombination. It cannot recombine. So whatever the gene you are giving, it will not integrate into the host. So they will replicate. So that whenever you want to isolate, you can isolate them. And whenever you want to store them, you can store them as a extracellular. They won't integrate into the... Yeah. Sorry, I'm not...
Here you guys are using DH5.
DH5 alpha cells. So mostly any lab, generally they use DH5 alpha cells for storing, for copying, making copies, everything. Easy for growth and
Like
easy for...
top 10.
Top 10 cells also. Fine, that is also. But this is one of the primary and easiest to grow. Top 10 is a eukaryotic or prokaryotic? Prokaryotic one. Then I think there is no difference. There are some vectors, some organisms, they store it in the eukaryotic. Depends upon the gene of interest. But here, if your idea is to store and to recover high copy plasmids, DH5 alpha works better. You're asking something? Yeah. Because DH5 alpha cells, they have a mutation in the recombinase. As a result, they cannot be able to recombine. Whatever the plasmids you are giving, they will not integrate into the chromosomes, nuclear chromosomes. So that's the reason, because whenever you want to isolate the plasmid, you should isolate. If it integrated into the nuclear genome, you cannot able to isolate, right? So that's the reason we keep DH5 alpha. So you will transform your plasmids, whatever the plasmids. We use heat shock method. You will be going to do the heat shock method. So you will be transferring whatever the vector. It doesn't matter. It will take up your vector and it stores. And whenever you want, you can put it in the LB media, grow it overnight. You can isolate the plasmids. So it acts as a storage as well as for multiplying the copies. Two purpose DH5 alpha is used. Anything? Fine. Okay. Then finally we will be going for the transformation using the electroporators. We use the electroporator today for transformation. So simple. Any doubt in the outline today? So first I will be giving you the theoretical explanation how the cloning will be done. So what is cloning? I think she might be knowing. Okay, I will come to you later. Let's ask the students. Last answer will be yours. From this side, I forgot your names actually. What's your name? Shamila. What's your name? Asmita. Yours? Pranati. Okay. What's yours? Subhashri. Yours, bro? Dashrath. Okay. Okay. Any of you, come on. Let's take who can say the better clear definition.
Means, simply making many copies of the same organism.
No, cloning is not making copies.
Other foreign names.
Yeah, make it very clear. Yeah, cloning is nothing but you are inserting whatever the fragment, whatever the gene of interest into the vector. That is called cloning. And it involves two steps. One is digestion. Digesting the plasmid, because plasmid has, morning I was telling you, different parts, right? In that multiple cloning site I said you, where you will be inserting your gene of interest. That multiple cloning site, for example, let's take, this is the vector which we are going to use today. That is PSL 18. It is well standardized for the Chlamydomonas for expression. It has PSA D promoter and multiple cloning sites are here. Okay, and this is the promoter, PSA D promoter, photosystem protosystem 2 promoter, followed by this is the multiple cloning site, and it has many restriction sites. You can see these are all restriction sites. Particularly this region, we can use whatever the restriction enzyme available to you and compatible with them, we can use them. So this is the vector. Enzyme is not present in the gene of interest. Why the second point is important? Why the restriction site should not be present in the genome?
It will cause the gene of interest and we do not get our
Yeah, product. Exactly. So the restriction enzyme you are keeping should be unique. It should not be present in the target gene which you are going to clone. So how you will identify what are the restriction enzymes present in your target gene or not? Is there any tool? So there are many tools out there. I use two tools. One is SnapGene. SnapGene, once you give a sequence, it will say what are all the restriction site available. As well as if you go for NEB cutters, if you give your sequence, they will be telling you list different 1 cutter, 2 cutter, 3 cutter, and there's an option called 0 cutter. So I will be showing you 1 second. So this is NEB cutter. This is one of the software, I can say. So they changed it. It's latest. I think Wi-Fi is a bit slow. let's take, I'm randomly taking some sequence and I'm pasting in the zero cutter. Okay, this is the dialog box. I'm pasting my sequence. Now you can see there, same thing I've done. Then I'll be submitting. So you'll be getting the list of restriction sites, right? So here the one which is selected is one cutter. So if you go for enzyme list, these are all these whatever they mention will have single cuts in your rest. It shows where it is also, but you need zero cutters, right? You select zero cutters. So these are all whatever listed, right? These enzymes were not present in the sequence which I have given. So you can select any one from this, but still you need to cross check whether that enzymes are present in the vector. So in this way you should select the restriction site, and you should design your primer in a way that it should amplify along with this restriction enzymes. Okay, now I'll be back there. So in this way you need to design your vector, and you need to amplify your gene of interest with the 5' end should be having the restriction sites. So then you will be amplifying and you will be digesting, like here you can see, no, we will be digesting both the plasmid as well as the gene of interest. So here also sticky ends will give better results. For ligation there are two types, no, blend end and sticky ends. So people always prefer for going for sticky ends, but in unfortunate situation you are not getting any compatible restriction enzymes, then finally you should go for the blend ends, but last option should be the blend ends. And then you will be going for the ligation. I will be telling what is ligation. Okay, I think here I haven't kept the slide. So okay, no problem. Ligation is nothing but you will be incubating the digested two products are there, no? So how you confirm it is digested or not digested, the plasmids? So no, you are digesting with the enzymes, but how sure that they are digested? Yeah, you should run on the gel, right? So you will be running on the gel. So generally undigested plasmids will have how many bands theoretically?
Undigested, single.
Yes, sir, telling single band. Anyone? Undigested plasmid should have how many? One. Wrong. Majority is wrong. Undigested plasmid will have 3 bands theoretically.
Like they shade.
They actually supercoil. 3 bands. You remember? There will be 3 bands. So generally, people don't try to run in the undigested plasmids because they will get 3. They will confuse. So you know plasmids are circular. So when you are going to run, there will be 3 possibilities. One is they supercoil and they become very short, compact. So they will be the least lower bottom, one band will come, and there will be a middle. That will be a niked one. Niked one means unfortunately some cut happen, one cut happen, they will become linearized. So linearized will be in the middle, and the top one will be the complete DNA what you expect. So when you run a normal without undigested plasmid, you will get 3 theoretically, but generally we will get 2 because the linearized one will be rare to get, but the complete open plasmid and the supercoiled plasmid you can see always. But when you digest now, how many bands you expect? If you digested, the digestion is complete, you should get single bands. If undigestion is there, then there will be 2 or 3 multiple copies. Then you need to identify which one is the right one. For that you should keep a control. For example, here in this case you are digesting with 2 restriction enzymes, right? But you got let's take, instead of single bands you got 2 bands. So how you analyze these results? What can you say?
So can you repeat the question?
You want to digest the same, like you are digesting the plasmid with 2 restriction enzymes. You expect a single band, but you got 2 bands. What will be, how you analyze this result? What you say?
Digested.
Partially. It would have been partially digested, or one enzyme would have worked, another enzyme would have not worked. So you need to keep negative controls. Like individual restriction enzyme, you need to digest and check whether both the enzyme can able to give digestion properly. And sometimes incubation time, if you increase the incubation time also, the results will be better. So you need to try different methods to find out which works better for you.
Yes, control means A DNA which was well digested.
Control means without digestion.
Yeah.
Control means without digestion. You will be loading then single enzyme. That will be a first treatment. Then enzyme 2. Then enzyme 3. Means enzyme 3 means dual combination. If both the single gave a single fragment, that means both the enzyme work properly. Then more likely the third one also should work. If either of these two enzymes are not working, then you can predict that which enzymes are not working. Okay. Okay. Any doubts here? Yeah. That's what I was telling. No, you need to go for the NEB cutter. It depends. It's not the particular enzyme you are going to use for all the experiment. Yeah, in this, the gene, right now we are not going to use any restriction enzyme. I'm just explaining how to do it. So if, for example, if I have a gene, let's take hypothetical condition. I am checking in the NEB cutter. Right now I was checking, no, in NEB0 cutter I got few restriction enzyme, and I will be cross checking. Let's take, I got a NEB0 cutter eco R1, and I will check here eco R1 is there. Okay, then I can put eco R1 in my gene of interest. Similarly, I will look for another restriction enzyme for the other side. So like this, I will be taking two enzyme and I will be amplifying my target of interest with those restriction enzyme. Okay. Then after digestion, you confirmed with the gel, you need to go for ligation. You need to gel elute, purify. Then you need to use the T4 DNA ligase. So what is the purpose of point of ligase? What does it does? Phosphodiester bonds, right? Means the DNA will come in close proximity because of the random molecular movement. But the sealing has to be done by diester bonds. So that will be done by the help of the T4 DNA ligase. And generally we keep overnight. We keep it at 18 degrees Celsius overnight. And the next day morning, what we will be doing, and let's take 1 microgram of DNA initially for digestion. And you will be ligating all of them, right? And you are going to transfer the next day all of them. But in the 1000, let's say 1000 microgram, hardly 1 or 2 plasmids would have ligated. It's highly probability based, right? Not everything will be ligated. So we need to find out which one has ligated, which is not ligated. So for that only we are going to transform into the DH5 alpha cells. So what is the advantage of transformation into the DH5 alpha?
You can make out express.
Yeah. If they are sealed completely only, they can able to express. If they are not sealed, they cannot able to express. So how we are going to select? We need to check any antibiotic resistance. Here in this case, ampicillin resistance is present, right? Oh. No, here ampicillin resistance is there. LAG-Z is not there. You can use LAG-Z also there, yeah. LAG promoter is there, no? We can use two things. Generally, if you want to use LAG-Z also, you can use to check whether insertion is there or not. So ampicillin resistance, once you transform into the DH5 alpha, you will be growing them in the ampicillin, LB ampicillin plate. So what if the colony appears the next day? Most likely they should be having the sealed plasmids with gene of interest, but still you need to confirm. How you confirm the plasmid has your gene of interest or not? Gel? Gel is not the first step. No, selection we are growing in the plate. Ampicillin is not a strong antibiotic. Sometimes artifacts also can grow. But how you are sure that your gene of interest is present in the plasmid or not? Yeah. How you confirm? You need to do some confirmation, no? Tracking?
First we check the OD.
Okay.
And then we will go for
What?
you can isolate.
Yeah. You can do isolation, but that is a little tedious job. Let's take you have 100 colonies in your plate. Your transformation is very good. 100 colonies in this. In this 100 colonies, you want to select which is having positive colonies. How you will do? You will isolate 100. 100 is tedious. You want to do it? You can do it, but it's a very tedious process, right? How? How? That's what I'm asking. Screening only how? No. You will do colony PCR. Simple. You will pick a colony and do a PCR with the G primers, same primers of gene of interest. And if you are going for any expression study, it is very important to check the sequence. You need to send it for a sequence to confirm whether the open reading frames are fine. Sometimes mutations may happen because you are PCR amplifying, right? So you should be very careful. Only if you confirm the gene which you have present is exactly the same, then you can proceed for the chlamydomonas transformation. So now we are going to start from, now you guys remember that you imagine that we have cloned. So we are isolated the plasmid, so we are going for DH5 alpha transformation. From now onwards, we'll be moving ahead. First step is you are going to do is transformation into the E. coli. Okay. So this is like transformation, isolation of the plasmid from the E. coli. This is, I already explained you. This step only we are going to do now. First the plasmids, we already have the PSL18 plasmids. So now the protocol book I given to you, no? Do you have that now? That day each group we are having, no? This is one I made it. Yeah, we can start. Yeah, of course. Means for inserting your gene of interest, right? Yeah, of course. Anything is fine. This is very primitive. TA cloning you can use, gypsum you can use.
Someone can get ice first. I get the ice tray.
Yeah, one ice box is enough. Give one. Yeah, one can go and collect the ice. No, vector is not DH5 alpha, sir. Vector is a PSL18.
Who knows where the ice machine
It's
is?
a cell, E. coli. In that only we are going to transform. Okay, then bring the
She's
plasmid. Okay, okay, once she brings.
take out the
Just follow this. What are the things required? PSL18. It's well used for overexpression.
LBNO is with you.
Sorry? Yeah, any vector you can use. Method is same. I'm just showing you with one vector. You can use it for the same for any vectors.
Sir.
And one more thing I forgot to mention. The ligation has different combinations. Vector to fragment, there is a molar ratio. How you will calculate? It's not like 1:1 ratio you will take. It will, based on the molecular weight, you need to identify what is the best matching concentration you will take. So for that, there is a tools if you really want to use. There is NEB cloner. So like NEB zero cutter, NEB cloner is there. So if you give your plasmid size base pairs, like PSL18 is 6,000 bases, and your gene of interest base also like 1,000, 2,000 you mentioned, and finally how much concentration of DNA fragment you are giving, 200, 300, then it will suggest different concentration, which one, 10:1, 3:1. So you can, it's like a random. You always suggest, I always suggest you to take more than 2 ratios, because it's completely a random process. It's nothing like 1:1 works. Whenever you're going for ligation, keep 2-3 different combinations.
How through the colony PCR?
Yeah, colony PCR is that day, first day we are doing some colony PCR in algae, right?
And how PCR helps to identify?
Yeah, that's what. What is the transformation means? Digestion and ligation, what we are trying to do, we are trying to insert our gene of interest into the vector, right? So our aim is the plasmid should have our gene of interest. So that's what, using PCR, we are trying to amplify again the gene of interest from the plasmids. If it can able to amplify, that means gene is present inside, right? So that's what we are trying to. What happened? Why you are changing? What's your name? You please come this side. What do you want?
Got pressed when I was ready.
Yeah, otherwise, if you want to sit, you can sit this side and write it. Laptop. Fine.
PhD data.
Yeah. Okay, any other doubts you can ask me now. We'll just wait for eyes. Once eyes is ready, we will go. You do agar-bacterium transformation?
For the dissertation,
Okay.
it was like a new plan. So
Okay.
I was trying to standardize.
But agar-bacterium for higher plants, no? Which organism?
Agmella odoraceae, Astraceae family.
Okay. So you're
It's a common
successful.
plant.
You're successful in that.
Like I was getting it, transformation, but it was what is it? Like we do gas expressions. P can be a vector.
Okay.
It has gas.
Uh-huh.
So the spots were like not uniform.
Okay.
I'm getting like spots. In case she will be doing the rest.
So it's like a part of your work, or it's,
Like she added my things with
oh, you already have a publication. Which journal?
like plant culture. Plants that PCTOC.
Please. Any of you has a publication? You. He's from a big lab.
Yes.
Well, green biochem. What is the journal? What journal?
For chemical engineering.
Okay.
Chapter in my one paper. It's very small journal. Environmental biotechnology.
Yeah, got it. Keep it here.
This is the vector.
Okay, I'll bring the DH. Okay, I'll bring the DH. Excuse me.
It is now.
Take that, it'll keep it nice. Yeah, one is enough. Oh. This is having what is the concentration, Shreya?
230 nanogram per microliter.
200. Okay, 200 nanogram. Pipettes 10.
This is 1 ml. 1 ml is there.
Okay. Need a gloves. Can you give me a gloves? MM. So who wants to do? One person will show anyway. So who wants to be doing that? You want to do? Who haven't done it till now?
Me,
You haven't
never,
done?
but I want to do. I never done it before.
So who want to give up among you?
No,
Okay. You want to do? Okay, let's first step is we have the protocol, no? Just I brought the competent cells. This is the competent cells. Competent cells preparation is you can get it from the protocol from the online. So there is nothing. You need to treat them with the calcium chloride. You need to rupture the cell wall. So it's a very simple protocol. Anywhere you can find it. Once you make the competent cells, you store in the -80°. Whenever you want to transform, you will keep it in the ice, allow it to thaw for one minute, and meanwhile you should transfer your plasmid of interest. The plasmid which we have is PSL18. We already isolated. It has some 200 nanogram concentration. So first, as it is mentioned, we have switched on the water bath. This is the water bath for heat shock. So 42 degrees Celsius, we need to heat shock. So first we need to take around 5 microliter of the plasmids. 5 into 2 means 1000 microgram we are trying, but that is too high. Maybe let's take 3 microliter, you transform. 3 microliter of the plasmids, you transform to the competent cells and then leave it for a while, 1 or 2 minutes. Then we need to go for heat shock. So heat shock is very specific time, 90 seconds I mentioned, no? 90 means exactly 90. If you keep for even little like 10-20 seconds extra, it will affect the cells. The efficiency of the transformation will get affected. So make sure once before going, you set the alarm. 90 seconds, time of keeping and taking out should be exactly 90 seconds. And then chill on the ice. Immediately you should chill it on the ice. And then you will be adding whatever the transform cell to the 1 ml of LB media. We will grow them in the 1 ml LB media. Why we are growing for 1 ml?
LP.
LB media we are growing, means immediately after the heat shock, we are transferring to the LB media and we are allowing them to grow for 1 hour.
1 hour you are making it to allow the insertion of the form.
No, that is done by heat shock. We are allowing to recover. The cell wall should be formed, no? Means the entire genetic material will penetrate during the heat shock process. So once after heat shock, we are trying to recover. No, they should recover. The cell wall should be formed. So this 1 hour will be enough because doubling time of E. coli is 20 minutes, right? So that's what. In next generation, they try to form their cell wall. That's the concept. So then you read it. Next protocol, what is it? I mentioned 1 hour. LB media, 37 degrees Celsius. Yeah.
After 1 hour, centrifuge cells. 6,000 RPM at RT for 5
Okay.
minutes.
We need to remove the supernatant and you need to pellet down the cells. After that,
Resuspend the cells with 100 microliters of LB media
fresh LB media. Again, we are resuspending the entire cells in fresh LB media.
and spread it on the LB plate.
LB plate with ampicillin. So that step that has an ampicillin, that's a selection marker.
Maintain the positive as well as the negative.
That you can do it. And then finally,
Incubate the plate at 37 degrees centigrade
overnight. Okay.
Cells
So we'll be splitting today and we'll be keeping it overnight. That step we'll be doing. And again, the same plasmid will be showing you the electroporation, how it has to be done. Two methods. One is transformation we are teaching, another one is electroporation, algal transformation in other way we can say.
Here we are not like getting enough interest with vector.
That's what I said. Today we are not demonstrating that. We are directly going into the transformation. Yeah.
Already prepared vector into
Yeah.
the ESL
Yeah.
class.
Yeah. That's it.
Sir,
What?
here we are using 1 ml LP and here we are using 100 microliters.
Yeah.
Why that difference?
We are going to spread it on the plate. We should not have more media. No, we should spread entire cells in a single plate. That's a concept.
Is there any alternative for colony here instead of colony here? There is no other method.
That's the simplest method. That's why we are using.
So
Otherwise, that should be the most simple media method. Otherwise, sequencing you should do. That is more complicated. Yeah, don't keep it outside too much time.
is there any
So you are going to do now. Wear the gloves.
so is there any specific ratio that how much of the vector should be transferred to how much of competent cell?
There is not a. For example, you can take 200 to 300 nanogram. That range.
Of vector.
If you take more vector, you will get more colonies, a lawn. So people don't like to have a lawn. So you need an individual colonies you require, right? So something lesser than 500 nanogram is more ideal for 100 microliter of competent cells.
500 nanogram for 100 micro.
Less than 500 nanogram for 100 microliter of competent cells. Okay. So now take pipette and set it to 3 microliter. Slow. Which side you know to turn? No? Set it to 3 microliter. 10 microliter tip box. 10 microliter tip box.
The thing is
Yeah. Tip boxes. Shell today, no sir, no need actually for today. Yeah, you can use this. So she's going to transfer 3 microliter. She has taken. Fine. 3 microliter of the vector and transform into the competent cells. Whatever. Oh wait, give that some more water. Give me that tip box. And again, molecular biology, everything. It's like a demo, so we are giving it in a crowd. But when you are doing it in your lab, try to be isolated, sterile, wear gloves, mask, everything. Yeah. So 3 microliter, take it exactly. Yeah. Fine. We can keep one more negative control. Yeah. And keep it in ice for 5 minutes. It should be kept in ice for 5 minutes. Shreya, can you take one more competentials? We will keep on negative control without plasmid. So technically, negative control should not grow in the ampicillin. Okay. Namrata, I took the plate, LB plate. So it means, I think why we are keeping it in ice, they would have been very frozen state, no? They should thaw first, and meanwhile the plasmid will get mixed completely inside. So when we go for heat shock method, it's more likely there will be a green in the middle. Okay, I'm coming. It's written at DH by take on. Later. Later. Okay. No. This is not at all light sensitive. You can do it in light, but isolation required. Sterility is very important for mold bio rather than light. So let's make this as a positive control. Marker required. This should be negative control. Label it, otherwise you confuse.
So one is with plasmid and competitive cells, and one is without plasmid and competitive cells.
Without plasmid. One treatment is with plasmid, another one without plasmid. That's it. So that you can see the difference.
Same competence.
Competence cells are same. Only thing you need to understand that the plasmid makes them to grow in the ampicillin plates. Sorry. So make sure always this is there. If it goes, then don't confuse, okay? So now we can go for heat shock. Who is setting the timer? 90 seconds. Timer is perfect. 90 seconds. 90 seconds. Means 1 minute 30 seconds. It can.
Stopwatch is there.
Otherwise phone watch you can use.
Stopwatch. Anyone keeps timer for 90 seconds.
This
Yes,
we
yes.
already done.
Yeah.
Continue keeping.
Yesterday, then
When did we do this?
What was?
90 seconds.
1 minute 30 seconds. Okay, you can see. Okay, you can set it for 1 minute 30 seconds, or you can watch till 1 minute 30 seconds. Anyway, it's fine.
Timer ends.
Okay.
1 minute 30 seconds.
That is 90 seconds, right? 60 is for 1 minute, right?
Okay, start.
Yeah, means immediately you transfer. Yeah. Once the moment she kept, you can switch on. Switch on. Other one also, negative control also. Yeah, switch on. So what is the next step? You focus on the chill them. You need to keep it in the ice. Immediately take it and keep them in ice. Then LB media. We need LB media. You can take the LB media here. Okay. But I need a Falcon, 5 ml Falcon, 5 ml FFO.
I mean, the vector
Two.
will consider.
Bring 2. No, unused.
Just one. Just hitting.
Competence cells.
Why are we doing this? No time.
Not no time. It's like yeah. Basically, time is limited, and moreover, even if I do it, requires a complete sterility. It has to be done in the laminar, not in a crowdy atmosphere. Even if you do in the crowdy atmosphere, it doesn't make justice for the work. So that's why I'm teaching you the theoretical part, and I'm directly moving into the
When have you done this? I don't know in the journal. Make these transformations.
last week. And once you can make years before also. Once you store inside, it's not an issue.
M13. M13.
Lambda.
What is the temperature?
5,
Like
4,
44 degrees.
Sorry.
This is an expression.
For how much?
It's 1 second. Son, just transfer it.
1 minute 30 seconds.
Transfer, keep it in ice. What you are asking?
The expression vector.
Yeah, it's expression vector. That's it. Okay. Use a
For us,
fine. Keep it in ice. Yeah. Let them for 5 minutes. Let them cool. Note down the time for 5 minutes.
you can see how am I mapping.
How?
T7, right? T7.
Yeah. That's promoter can vary anything. It's not an issue. Means this is like we purchase this promoter vector. It's not we made it. We purchased from Clammy Resource Center. So
1 ml.
nothing. Any promoter can be used until it's going to be working for you, right?
No.
The main idea for it's like for any overexpression we can use PSA-D is there. It's a strong constitutive promoter, and it has been widely used in many chlamydomonas like research. Earlier papers were there, so that's what we prefer to buy this plasma.
E. coli is 20 minutes now.
What am I?
He's asking if 1 hour it will go. So E. coli takes 20 minutes.
Yeah.
40 minutes.
20 minutes. Dividing is 20 minutes. Okay. 5 minutes. Let's keep. After 5 minutes, what you have to do? You need to transfer 1 ml to this.
We brought to the
Yeah. You label which is positive, which is negative. You can label it treatment. Another one is negative control.
Like when I did internship, they were doing silencing, RNA silencing. So they have done gateway.
Great. I use gateway for some complicated, like gateway for guide RNA. When you are targeting multiplexing, they used to say multiple segments you need to incorporate. No, that time this method is very difficult. Restriction, digestion. So simultaneous incorporation of multiple fragments, gateway cloning is a bit advanced to understand. But once you establish it, it's easier.
Nitrogen peanut plant.
Okay.
So
That part.
Yeah. Anything?
I am confused in between these two.
What is it? A confusion? Tell me.
Like which tube is containing vector, which tube is containing competent cell, what
Yeah. Only one. Wait. Only one thing we changed. 2 tubes we are having. Both the tubes are having competent cells.
okay.
One is we added the plasmid. That will be your actual
Only
sample.
one.
Only one tube we added the plasmid. That is your actual sample. Another one we kept as a negative control. Negative control we didn't add any plasmids.
Negative control means?
Negative control means there should not be any growth. Means we are believing that the growth of the cells in the ampicillin plate is completely because of the plasmids.
Okay, that's only competent cell only. We have
Yeah.
not added the vector.
Vector is the difference between the negative control.
Vector nahi.
Yeah. For any experiment, you keep one positive control, one negative control, right? So in this case, negative control is without the plasmid.
Okay.
So we are transferring the vial with competent cells and plasmid to heat and ice.
You can keep the both we kept. Means everything should be the same, right? Positive control, sample and the negative control should be treated with everything the same. Only thing, it does not have a plasmid. So that you can say the sole difference between them is because of plasmids, right? For example, if you are not treating the negative control to temperature, then sometimes the cells, you have a doubt. Maybe because of temperature, they may not grow. So everything you should follow the same pattern, except the plasmid addition. Remaining everything you should follow. Added 1 ml, 5 minutes over.
5 minutes more.
Okay, fine. You just add 1 ml of LB. You label 1 as a negative control, no?
C is a negative and P is a negative.
Okay, fine.
The competent cells with the vector, they are in the plastic, and we have kept them in the water bath. So the 44 degrees enough to
Penetrate.
like enough.
Yeah. It's well.
Because the plastic is also there.
Plastic?
Plastic, no, this ependophthalmic tube.
No. Anyway, cells are present inside the ependoph only. We are not blocking in between.
No, I mean the heat is enough. So that
Yeah,
passes
it is
to the
enough.
plastic.
It's well. Fine. 45 to 60 is more than enough.
He's saying that we are using plastic tube.
Yeah. I can understand.
There is
Enough.
Is there any chances of plastic leakage to the
So he's telling temperature is enough to transform or not.
So there is a barrier also.
Yeah. Still it's enough. We tried, it worked. So in your case, if you try, if it doesn't work, then you should try it for higher time. But if you're going for higher time, it will kill the cells. They are very sensitive to temperature. So 45 to 60 is more than enough. I'm giving 30 seconds extra. Add the respective cells to the respective tubes. That is very important.
Next time.
You
broth, no? Incubate 1 hour, 37 degrees with constant.
No, she should transfer that cell to this. Yeah, negative control to the negative control tube.
that only competent cells and vector is added anything?
No, you can use 1 ml tube. Entire sample you transfer. What do? You take 1 ml, transfer like little to the competent cells. Okay, entire thing, and let's take treatment. You are going to start. Take little media from here. Don't touch anyone. It will contaminate. Take little. Okay. Keep it open only. Keep it here. Which sample? No, sample. This is negative control.
Okay.
You don't confuse. Mix it
Understood.
and take the entire thing. Slowly. Gentle. Be gentle. Take entire thing slowly.
What is added?
We added 1 ml of LB media, mixing and transfer to the bigger tubes. It should grow in them for the recovery. Slow. Treatment, right? This is treatment. Okay, now similarly you change the tips. Yeah. Don't use the same tip. You will get contamination. Now you do the same thing. You take some ml of media. No. Tip. Huh?
Sterile media.
Yeah, sterile media.
Competent cells and the vector.
Hmm.
And negative control is
Without plasmid.
without plasma.
Same competent cells without plasmid.
And positive control.
Positive control, if you have any other plasmid, you can use it. At least negative control is very important to check whether contaminations are there. Sometimes ampicillin, as I mentioned, they are weak antibiotic.
And after this, we will do plating
Yeah. Finally, we will be plating it,
on ampicillin.
ampicillin plate, and we'll keep it overnight. Tomorrow morning, you can see the colonies. 1 day it takes to form the
In
colony.
this colonies, what we do?
We'll go
PCR?
again. We'll check the colony PCR to find out. That we are not doing, but I'm telling it. You should do it to confirm. For now, we are going to. I'm just teaching you the transformation into E. coli. That one step, and another one transformation into the algal cells. These two transformation I'm teaching you.
In that large tube, there is LB got
Transferred, right?
Yes.
Now we need to grow them in 37 degrees Celsius for how many?
37 degrees.
1 hour.
Hour at 37 degrees.
Shreya,
Constant.
need to keep in Narendra's lab these two. Keep these two in the incubator. 37 degrees Celsius. You just say E. coli.
Static condition.
It's shaking.
Chill condition.
But it's occupied, no?
I don't think so. It should be.
Fine. You can keep it there. 37. It is 37 or what? If you want to remove, you can keep it here. Then set 37 and keep it here.
The temperature maintenance will be done for this
Which one?
incubator shaking.
Yeah, 37 is very important.
And on the next day, we have to isolate the plasmid.
From these plates. From these plates. We need to pick a single colony, inoculate in the LB media, overnight again. From the LB 10 ml media, we will isolate the plasmids and again go for algal transformation. Okay?
We will transfer the plasmid to the algae.
Yeah.
So now you have already isolated.
Isolated plasmid, so we are directly going. So I am bypassing the steps. That's it. Any doubt in the steps?
So from this, gene of
నువ్వు
interest,
చేసేటప్పుడు చిన్న ట్యూబ్స్ లో ఉంది కదా
Sorry.
what you are doing here?
దాని ఫస్ట్ కొంచెం ఎల్బి మీడియా వేసి
Right now, that's what, sir, we are transferring in the plasmid. For example, your gene of interest has been cloned into the vector. So now you want to multiply those copies, right? One or two plasmids only you have, but you want to multiply them into hundreds, 200s, more concentration. For that, you need to transform into E. coli. Because E. coli doubles faster, makes multiple copies, right? So that's why we are transferring into E. coli.
what we will do.
That's step one.
So that's for the components also.
Now this algal culture, what we are doing in algal culture, gene of interest.
Yeah, finally, the entire aim of this program in this molecular biology is to transform your gene of interest into the algae. For that, we are doing all these steps right from RNA isolation. We will be isolating what gene you want to amplify. You need to amplify the gene that we did on that day. After amplifying the gene, you need to clone into a vector.
44 degrees heat
That we are doing. We should have. I explained you theoretically how it has to be done. Then we need to isolate, bulk it up, that cloned plasmid, and finally we need to transform into the algae. It's all a single sequential experiment has to be done.
ఒక పెద్ద స్పెండ్ తీసుకుని
It's up to you. It's not necessary that it should be very fast, but you can complete it if you work hard.
What hard, how much time?
చేసి సస్పెండ్
It will take a week.
ఆ
That's it.
తర్వాత 1 ml మొత్తం
పెద్ద దాంట్లో
up to algae.
ఆల్రెడీ చిన్న వాటిలో
Yeah,
ఉన్నాయి
yeah.
కదరా.
Algal and after transformation into algae also, it will take big to grow. Algae grows slower. From that also you need to do screening, multiple screening.
ఆ తర్వాత పెద్ద దాంట్లో ట్రాన్స్ఫర్.
So
మనం
we are
పెద్ద దాంట్లోకి
desired-
ఎందుకు ట్రాన్స్ఫర్
desired body
గ్రో
is
అవ్వాలి కదా.
Yeah.
చిన్న దాంట్లో వేసినా కూడా
Ready? Chloroplast.
Bro,
చిన్న దాంట్లో వేస్తే
nah,
గ్రో
Vijay,
ఉండకపోవచ్చు.
we can go for break now.
And the
Arrange it for we have 1 hour break.
centrifuge
1 hour time.
both are centrifuge tubes, right?
We are doing chloroplast isolation transformation today.
Yeah, the final after this plating, I will be showing you how to do the transformation using
And
electroporator.
tomorrow.
Mall bio ends today.
Okay.
BNP,
Tomorrow something.
BNP,
That
Thylacine,
my
yesterday we-
lab members will help you.
Yesterday Thylacine we have isolated.
Tomorrow we'll run.
Then
Tomorrow we'll do, sir. Yeah,
BNP.
tomorrow we'll do. So yesterday, how was it? I left in between after Thylacine. How did you see the peaks?
Yeah, very nice. They have
Ah.
just
We got very nice PS2 and PS1.
That's 77.
You got 2 picks. Jyoti Bhaiya, they got a second pick also.
No.
Better than us. Lower. We did it or yesterday what? That means because of the higher concentration. You guys are so lucky. Because I did it like last week. I didn't got that good.
7 micrograms.
You have a photo? Jyoti Bhai, you have a photo? Okay. So you plotted also, you guys plotted. Sir.
Not plotted, but
Today they will show how to.
Oh, you are very late yesterday. When I was leaving, it was 6 something.
20 minutes.
But CD is a very good technique. And with liquid nitrogen particularly, you should be very careful.
Not serious.
77, sorry. 77. I was confused. I think almost our experiments are near. BN, tomorrow BN. After BN, tomorrow it will be completely BN. It takes a long time for running. And after BN, I think HPLC, one more experiment is there. CD is not there. BN is there.
ఇప్పుడు షేకింగ్ ఇంటిగ్రేట్ మళ్ళీ తీసి మళ్ళీ 1 hour సెంటిగ్రేడ్ ఆ తర్వాత ఏమంటే అప్పుడు రాస్తాం.
Nice. I think Jyoti Bhai, we need to increase our concentration. I think 5 is not working. Next time we'll go for 10. Yes,
one competence
but should not create that much difference. So what you understood? Why yesterday everything you understood about 77K? What it is done? Why it is done?
PS1, PS2.
PS1 and PS2. PS1 particularly. We can't see the PS1 bands in the normal.
That cryo
At low temperature, very low temperature, you can see that.
using the quartz.
Yeah,
Cuvette in the sense that is an NMR tube.
in high temperature it can
very quad zone.
Quartz is NMR tube. Narrative,
Oh,
nuclear magnetic resonance.
yeah,
tube.
yeah.
NMR, you biologist might not be knowing. No?
Yeah, I haven't used it. That's
That's
right.
why we are seeing the biology of the
Yeah.
రాసుకున్నా ఉంటా
But NMR, that is NMR they are using actually for spinning.
Okay,
They
okay.
are NMR, we have to our synthetic in the laboratory they are synthesizing the compounds.
Okay.
For synthesizing compounds, they will see that they will remove some groups are there. They will
Okay.
then they will put and they will see the NMR.
Oh.
Scaffold, what is the NMR protein? And the radiations, then they can see the peak, the magnetic resonance, nuclear magnetic resonance.
I'm not that good in that side. NMR. Yeah.
This is also there are 2 parallel things.
Yeah.
That is chemical characterization.
Yeah.
Biological characterization through protein.
So morning class, you guys understood something?
yes
Chloroplast transformation, they understood something?
Yeah, you are nice.
Oh. It was like a last minute preparation. I was given a last minute. So I was in a hurry to make.
that much nice you're
Well,
presenting.
And the topic is very small. It's not a big topic. Very narrow down topic.
These are very things for you. That,
I used to do. My seniors are there. Few are right now not there. I am just doing CRISPR.
1000 RPM, 1 hour.
1 hour. I made that side because
It's okay.
It's fine. Okay. What is the next step? Fine.
Pre-suspense cells with 100 microliter of LB media and spread it on LB plate with syringe.
Yeah.
100 micrograms.
LB plates are here. Okay, but it again I'm telling this has to be done in the lamina. For our convenience, we are doing it outside. It's a E. coli. Definitely, it should be done in lamina. But for now, we are doing it outside. Okay.
No, we don't have laminar.
We'll just you have a
Here
spreader.
we don't have laminar.
Spreader.
Even in the lab is not there.
That is not working. We will go down. We have culture room. That is there. And in the laminar also, these many people should not stand. There is no point. So almost we will spread it and we'll keep it in the 37 degrees Celsius, right? Overnight. And this step is done here, and then we'll go for algal transformation. We need to little bit speed because that electroporation we need to go to other lab. Before they leave, we need to. Otherwise, tomorrow. I'll show you once again. Not that charged.
Move like this now. It will come noise.
Okay, let me brief you about the transformation, algal transformation. Just can you see the screen there? Oh, it's not moving.
Different time away.
And okay, yeah, now we can. So electroporation, how many of you know the principle of electroporation? Morning I was talking you about two transformation method, right?
No, but plus or minus voltage, some particular voltage that cells pores will be open for that.
That's what I said. Morning I haven't talked about the electroporation.
That the GN means the vector will be.
Yeah. So why we are using here, now I am focusing on the not chloroplast transformation, nuclear transformation I'm doing. For nuclear transformation, you can use electroporation. So here, what is the principle? As she was telling, we will be giving a short high voltage pulse, electric pulse, that is going to break the cell wall. So by this, we are creating a permeable pores, and at this time, whatever the extra chromosomal DNA, in this case plasmid, will be allowed to get inside the cell. And once they are inside, how the recombination happens in terms of nuclear? Is it homologous or non-homologous? Random means? Non-homologous. Be prepared for this kind of questions. So non-homologous question. So non-homologous recombination will happen. So randomly, they will just incorporate it into the position. So then you need to screen using a PCR where they have been inserted. For example, you want to interest in a particular gene, then you need to amplify the gene near to the gene. By seeing the shift in the bands, you can confirm, okay, out of the 100 colonies, 5-10 colonies have a insertion at the particular colony, particular position. You need to screen colonies which are very specific to the insertion. That's your target, right? So that's the image you can see clearly. When electric pulse are given, they used to break open the cell. As a result, based on the inverse polarity, they used to enter whatever the genetic material they will enter, and then whatever mechanism you can even send Cas9. If you are given Cas9, they will tends to break the cell. Double stranded breakage will happen, and if you are not going to give any Cas9, then random incorporation will happen. It's up to you whether your interest is just a overexpression, then Cas9 is not required. But your interest was to create a mutation, then you need to send the Cas9. Fine. This is the protocol. So this will be the instrument electroporator, and this is the main instrument, and this is the chamber where you will keep your cuvette. Okay. And this is the processing. We will be taking some 100 ml of cell roughly, CC1, like a well grown, around 0.6 OD, and we will be pulling down, harvesting, and that cell we will be resuspending in a buffer, max efficiency transformation reagent. That's a commercially available reagent that used to
Kit only it will come.
no, it's separately you can get it. This is a reagent. The main purpose is to remove the cell wall.
What is the maximum efficiency reagent? What is the composition of that reagent?
That is commercially available, sir. I am not sure exactly about.
Sometimes they should mention.
No, you can check it. It's an in vitro gen product. Majorly they use it for the cell wall removal. Whatever they may have enzymes to remove the cell wall or something. You can check it, in vitro gen max efficiency reagent. You can type it. You will get it. Okay, so you will be washing that with twice with the max efficiency reagent, followed by you will be incubating your plasmid. In our case, it's a PSL18, right? We will be incubating them, and then we will go for the pulse in the cuvette, which we have a specific cuvette, and we will be giving a pulse that is 300 volts. Very high pulse, 2 times we will give, and this pulse varies to different methods. So this is like 300 is generalized protocol, and there are multiple. If there is a different instrument, different pulse. Cannot set the program for 2 pulse. Simultaneously we can set only 1 pulse. So according to this protocol, I have given 2 pulse, 300 volt for 8 milliseconds. So milliseconds means you can measure how short span. 8 milliseconds you are going to give 2 pulse, and immediately after that you need to transform them to a tap media, and it has to be kept in dim light overnight, and the next day it has to be plated in the tap plates. Okay, this is the protocol we will be doing immediately after this. Okay, tap is nothing but tris acetate phosphate. Generally we grow the chlamydomonas in the tap media photoheterotrophically. We supply the acetate, tris acetate phosphate, and finally we will be confirming the mutants. This is the plate which I generated. This is again negative control, positive control, and this is the actual plate. In terms of positive control, we can see colonies, whereas negative control there is no colony. This is the transformed cells, and we will be confirming using either PCR, RT-PCR, and localize. If your vector has any fluorescent tags, you can visualize them in the confocal microscope. Okay, fine. Let's stick to the E. coli transformation. Now what's the step we need to do? You need to remove the media, right? Yeah, who is going to do that? You remove the media and add 100 microliter of fresh LB media. You want to do?
Need to
What is the step?
resuspend with 100 microliter of.
Yeah, use this as a, that's what you remove. Use a pipette, 1 ml pipette. Use a 1 ml pipette, remove the entire media. Only the cells will be at the bottom, no? Without disturbing the cells, you remove only the media. Use 1 ml.
Discard, sir?
Yeah. Only supernatant, only the media. Don't disturb the cells. Remove entire media, complete media. Fine. Now you another
A bit is there.
fine. Otherwise, you will disturb the E. coli cells. Change the tips here. Can see. She is good in handling the pipettes. See? She should be very slow in releasing the pipettes. Should not be harsh. So now you add 100 microliter. Maybe you can use the different. Use this. Yeah, 100. No, that's it only. Just pull it, push it. No, here on, here only. What happened?
He thought it was chloro.
I thought
I didn't know it was written.
Oh, no. This is nuclear transformation. Only difference is both are similar. Only difference, the transformation method, there we use ballistic. Here we use the electroporation.
And we media 100
Yeah.
Should I get
Recess. Yeah, completely solubilize it.
I thought chloroplast.
schedule it is for time?
Okay, maybe initially, but almost similar. Nothing. Because of the instrument, we will go for this. But apart from this, you don't feel any difference between them. Only the instrument, how you are transferring them, that's a major difference. Yeah, fine. You go for the other one.
Ballistic here.
We don't have. We don't do actually chloroplast transformation in this university. If you want to do, we can look for ballistic and PEG mediated. We can do PEG mediated. Depends again, if you really want to do it, you can do it. No, it's a growth media.
Chloroplast in this lab regularly we used to do.
No, not regularly. Whenever we have some, it's not regular. No. Once we generate, then we'll. Chloroplast transformation we haven't done. Nuclear we do. Nuclear transformation.
They're working on photosynthesis.
Yeah, but it's not necessary that only chloroplast genomes is located in the nucleus also. That part also we work. Because already chloroplast related genome already well characterized.
Place where
Yeah, it's a growth media where you grow, no? Yeah, sucrose is just what can I say? Extra carbon source. It's done? Then who want to split it? You did it already, so don't come again. Let's go. Give a chance for other guys. Any of you want to plate? You want to plate? Huh? So this is the plate, and again I'm telling you have to do it in the laminar. So take 100 microliter. So in case if you are taking treatment, you mention control or treatment, whatever sample you are taking, and use the pipette 100 microliter. First you aliquot into the plate above it, then use the spreader and spread it. That's it. Okay. But you carefully note down which sample you are taking. Is it a negative control or positive control? You need to label that. Which? Yeah.
Remove all the media.
Tomorrow you can see.
Tomorrow you can see.
Same media.
Fresh media here.
Don't speak much and you be fast when you are opening. No. Particularly after opening, you don't keep it open for a long time as we are outside. So be fast. First you remove the plates. That's very important. Okay. And this also antibiotic added.
Out.
Sorry. Because you are telling when you will add that. No. You should not expose this much. You should. Right, I'll tell you. What I will tell. Give me. Anyway, I'm doing it outside, but still. This is negative control, okay? I'm taking negative control. Okay. Even if you are doing it in the laminar, open this much only. That's it.
So that's also negative only, right?
Yeah, this is negative.
I mean,
We need to label it. I can label it later. Wait. Now you cannot invert. Okay. Fine. That's it. You can discard. 100 microliter. Yeah. Now you label this. This is negative control, right? Okay. Yeah. You have one more plate or that plate? Show me. Media is very less. This is better. Use this plate. The way I added, similarly you add it. I will give you the spreader once again. Use this. Not touch it down. Very gentle while taking it up. Close it. Discard that. What happened? It was not there. Okay, leave it. Keep it. Oh, it was there. Fell down or? Okay, fine. It's not. Yeah, spread it. Spread it like how I did it inside. Don't give too much pressure. Yeah, fine. Fine. Throw it there. Yeah.
This one is
We need parafilm. You
have to
need
label it.
sample. Make it a sample. Where is the? So I need a parafilm. 2 parafilm. Here. Scissor. Here also we have. Cut it fine like this. This size. This size. Wait. This size. Whatever. No, fine. No, not this much big, sir. Fine. That is enough. Yeah. More than enough. Yeah, someone want to grab it?
Fine, it's okay. It's okay.
He won't show. He will only do. Just invert it.
Stretch it.
Stretch it.
From the beginning, from
Yeah.
starting.
Stretch it. Be fast.
Don't you over.
Yeah, same. Keep it from there. Slow. Need to join that. Okay, LB sample. Okay. Still, it's not sealed properly. Yeah, that. Okay.
Open. Take. So take.
Give me.
Yeah,
Give me.
salty.
It's fine. Let's work. What? She's afraid that it will spoil the sample or something. Okay, now it has to be kept in 37 degrees Celsius overnight.
You're keeping?
So yeah, 37 degrees Celsius. That's it. Okay, now we'll go for algal transformation. Okay, so I have already collected the cells. 20 ml of cells I have collected because this is OD, it crossed above 6.6. So the cells are already more. So I have taken less volume cells, 20 ml.
What OD? The
0.6 is the ideal. 0.4 to 0.6 should be the ideal OD. And if it is 0.4, then you collect more cells, 100 ml cells you need to collect. Whereas this is above 1, OD above 1. So I reduced to 20 ml. But I suggest you, because we didn't specifically keep the culture for transformation. That's the reason we are using the old cells. But when you are going for actual transformation, I suggest you to keep a fresh cell and always stick to the OD of 0.4 to 0.5. Keep 100 ml cells, stick to 0.4 to 0.5. That will be the best OD where the cells will be in the mid log phase. If it is like old cells, then probability of dead cells will be more. So I'm telling you before itself, when you are planning for actual experiment, try to be mid log phase. So 1 day later. So yesterday it should be 0.6, but it was today. So 1 day old. So but when you plan, exactly Chlamydomonas 0.6, 0.4 to 0.5 will come in second day, end of the second day. So something around, I can say 35 hours something, it will come. So you plan your experiment in a way that you will start the transformation at 0.4 to 0.5 OD. So now let's go for the algal transformation protocol. I think we need to harvest, wash. No, I elected this one only. So first we will be harvesting. We are going to harvest this. Wash twice with the tap max transformation efficiency, right? Okay. We need to harvest them. Come, I'll show you how to do the harvesting. Okay, Ragendra is there. Help Ragendra. Put it for centrifugation. You know how to use the centrifugation, no? Set it low temp, like low speed, low RPM, around 3000 RPM, 5 minutes.
This you are at.
1 minute. So yeah. 1.
Wait.